Hello, everyone. Good afternoon. Thank you so much, everyone, for coming. It's great to look out and see so many friendly faces on a Saturday afternoon. I'm Cassie Adams, Care Services Director with the ALS Association, Oregon and Southwest Washington Chapter, and this is our annual ALS Research Symposium. We have a lot of really great speakers for you today and a Q&A at the end. Just a few housekeeping things to take care of before we get started. If you're interested in connecting to the Wi-Fi on your phone, it's a open OHSU guest, and there's no password. If you have any questions, you can flag one of the staff members down to help you out. We have some cue cards or cards on your tables. If you have any questions that come up through the presentations, we are not going to be taking any breaks, and we're going to have a Q&A panel at the very end. So feel free to write any questions down as they come up, and you can give them to some of the staff members to be asked at the end. We also will have roaming mics, so you can ask questions as they come up at the end as well. For those who are live streaming at home, unfortunately our question link is not working, but if you have any questions about the presentation as you're watching from home, feel free to email myself or one of the staff members, and we will do our best to help get you answers at a later time. And lastly, very important, snacks and refreshments are at the back. And we encourage you to get up, stretch your legs, go out and use the restroom if you need to throughout the presentations, as we won't have any breaks. And restrooms are just right outside of this room, and you'll make a left, and there's men's and women's located right there. We also have additional restrooms on each of the floors, so you can take an elevator down if you need to. I would like to thank our sponsors today. Thank you to Hayek Medical, Ability Center, Mitsubishi Tanabe Pharma America, ITF Pharma, and Permobile. And also a huge thank you to Oregon Health Sciences University for helping us to put on today's event. And a big thank you to Providence Brain and Spine Institute for being here and for the clinical research that they do. Without further ado, I'm going to bring up our Executive Director, Lance Christian, to introduce today's speakers. Thank you.
Good afternoon, everyone. Can you hear me? Great. Whoops. There we go. Great. I'm Lance Christian. I'm the Executive Director of the ALS Association, Oregon and Southwest Washington Chapter. Our mission is to support the efforts to find treatments and a cure for ALS, while we also provide compassionate care and support for families living with ALS. We serve the entire state of Oregon and Southwest Washington, and as part of that effort, a nationwide effort, we're part of the National ALS Association. And through that community, we support and fund ALS research. The ALS Association Treat ALS program is the largest nonprofit funder of ALS research in the world, last year funding close to $20 million in ALS research. And how that happens is support from across the country, including from here in Oregon, gets pooled together to fund interesting and unique science for translational research and basic science purposes wherever the best science is being done. And so literally at any one time there are around 130 different research projects being funded across the United States, Europe, Asia, South America, and even a recent grant in South Africa. So you can say when you support our research efforts, the sun never goes down on our efforts to find a treatment and cure for ALS. Locally here, I'm really proud to share that besides our normal yearly contributions, since 2012, our chapter has rallied to provide over $1.2 million in additional funds for ALS research, including supporting a large research grant to Dr. Joe Beckman, one of our speakers today. So all of this means that really since the Ice Bucket Challenge, and I know some of our speakers will talk about this, there's been a phenomenal amount of activity in the last number of years. My tenure, I've been with the ALS Association 16 years, and I'm more excited now than I've ever been before because we've seen amazing amounts of new researchers, cross-pollination, more papers being presented, more research happening than ever before. Since the ALS Ice Bucket Challenge, 5 new genes have been discovered, and the energy is pretty remarkable out there. You know, we used to come to conferences and the neurologists would be discouraged, but now what I hear is hope. And so with that today, we're excited to have some amazing presenters. We want to thank them for their time and their dedication for their work. And I'm going to present them, introduce them, and then be thrilled to have the opportunity to learn from their experience and the cutting edge things that are happening out there in the world. So our first speaker today is Dr. Shafiq Khurram. He is the Associate Professor of Neurology, School of Medicine at Oregon Health Science University. He did his residency at Beth Israel Medical Center, Albert Einstein College of Medicine in New York. He did fellowships at Mayo Clinic and Mass General Brigham and Women's in peripheral neuropathology and neuromuscular medicine. He's a member of NEALS and the American Academy of Neurology. And I want to share his center participates in many clinical trials. He's a frequent collaborator across the United States and speaker at other organizations and other chapters. So please join me in welcoming Dr. Shafiq Khurram.
Okay, thank you, Lance. I'm going to first start by thanking Lance and Cassie for the effort that they do and for putting this symposium. I think it's very important for our patient and caregiver to listen and see what's going on.
Look, see where you're at.
Okay. There you go. I have to stand here. And also I can see myself. All right, that's very embarrassing. Thank you. So thank you for doing this, and thank you for being able to invite outside expert. Especially a big thank for Dr. Harms. You know, I just realized, and my wife reminded me today, that tomorrow is Father's Day, and Matt had 3 daughters, and he came here, and I just realized that you miss spending tomorrow with your daughter. So thank you very much. And also thank you for Dr. Anstrud, who I was first assigned to give an hour talk, and I was like, I cannot talk for an hour. People are going to fall asleep. So I decided to invite Dr. Anstrud, who's going to fill another half an hour, and he's a great speaker, and he's going to wake us all up after this. So this year, Lance asked me to talk about frequently asked question in patient and people with ALS and their loved one. And you know, I tried to remember a little bit what questions come up frequently. I also asked Ivel, our awesome nurse, what questions do patients ask her? And she's sitting there. Thank you very much for coming. And we came up with a list of different questions. Of course, unfortunately, ALS is a terrible disease. And one of the biggest questions is, what is the prognosis and what's going to happen next? So we're going to try to talk a little bit about this, although it is a kind of a delicate question, and I do feel a little bit uncomfortable saying it and going in all the details. But I think it's important for people to be able to plan their life and talk with their loved ones if there are things that they want to do on their bucket list to be able to do it. And I think one of the major implications, about 3 years ago, one of my patients with ALS unfortunately passed away, and then I called their spouse and asked, you know, how was your experience? What could we have done better? And the main thing they said was, I wish you kind of told us better and stressed out more how much time we had left, because we would have loved to do some stuff. And although a lot of time we do discuss the prognosis early on in the disease, it's when we announce the diagnosis, it's a flood of emotion, and it's hard to really remember every details that we say. So that stayed in mind, and I try now every time that I see somebody with this terrible disease is to kind of just talk about how things are going, how the plans are going, how the progression is going on, and so forth. So the next question is, of course, the medication for ALS. Unfortunately, there's no cure yet for ALS, but there are a couple of medications that I'll talk a little bit about. Supplements is a big thing, and whenever we don't really have a good treatment, supplements come in mind. Diet, exercise, as I mentioned, Dr. Anstrud is going to be talking about those. Stem cell therapy is a very frequent question, and then we'll talk a little bit about available clinical trials here in Portland and Oregon that if somebody's interested can participate in. So prognosis, this is a graph that shows you a group of people with ALS in a single ALS clinic, how they progressed over time. And each of these lines is a patient. And as you can see, it's highly variable. So some people, unfortunately, progress within 12 months, and some other lived 84 months and longer. And we know in our ALS clinic that we have patients who unfortunately progress over 6 to 12 months, really rapid progression. And we have other patients who live 10 years, 15 years, and even longer. So ALS is a heterogeneous disease, and really, we always say that the average is 3 to 5 years of survival, but this is an average, so this is combining hundreds of patients, and it really varies from one person to another. And the reason this varies is we know that, for example, age, the younger people are, usually they tend to do a little bit better. Site of onset, so if the disease starts in the arm or in the leg, they tend to do a little bit better than if they start with the what we call bulbar, which is the speech and swallow muscles. Delay in diagnosis. So this is a good thing. So if the diagnosis is delayed, usually it means that disease is progressing slower, and that usually tells us it's a little bit better prognosis. If the breathing is involved early on, that's not a very good thing, because this is really what is the main reason why the disease progresses. The presence of dementia is also not a very good thing. Typicality of ALS, so if there's anything that is atypical, that doesn't really look a lot like ALS, that's usually a little bit better prognosis. Weight, people who are malnourished and underweight don't do as good as people who have normal weight or are a little bit overweight. And then finally, genetic mutation. We know that certain genetic mutation can cause rapidly progressive disease, and some other cause a kind of a slower progressive disease. And Dr. Harms is going to talk a lot about the genetic of ALS later on. So in order to kind of better understand the prognosis and try to do prediction model, the European Network to Cure ALS, which is a group of ALS center, kind of the equivalent of NEALS here in the US, came up with a study. They collected data on thousands of patients and tried to divide patients into kind of slow progressor and rapid progressor, and come up with a way to try to understand a little bit how the disease progresses. And they come up with a tool. It's an online software that can, if you input data, it can kind of give us an idea how things will progress. And it's not a perfect tool, and there's no way to predict 100% accurately how things are going to go, but it gives us a good idea. For example, you can see in this, when we input the data, it gives us kind of a survival curve. And there's a good standard deviation, if you want, so 24 to 36 months, so 12 months is a lot, but it gives us an idea how things are going to go. And if patients are really very interested in knowing their prognosis, we sometimes use this tool. Now, going to the cause of death, as you probably know, ALS most of the time will progress to involve the breathing muscles. And this is unfortunately how things progress really fast. And the involvement of breathing kind of impairs not only survival, but quality of life. And this is one of the reasons why we really emphasize the use of non-invasive ventilation. And probably one of the best intervention that we do in our clinic is do a good teaching about breathing and BiPAP and the importance of starting that early on. It is uncomfortable, having air being pushed in your mouth and throat. It's really not a good thing. But I think if we start using it maybe a few minutes, and then a day, and then a little bit more later on, and eventually we can use it for a couple of hours or even longer, that would be a good thing. It does affect quality of life. If your brain is lacking oxygen when you sleep at night, your waking up tired, you're not in a good mood, you're fatigued, you don't have the energy to do a lot of things. But if you are getting the oxygen you need during your sleep, you usually tend to do much better. And not only the quality of life is much better, but the survival probability is increased as well. Now, the next common question, medication and supplements. As most of you probably know, the FDA currently has 2 drugs approved for ALS. And these are the drugs that also are used elsewhere in the world. Riluzole, which has been with us for over 20 years now, and edaravone or Radicava, which has been approved a couple of years now in the US. Riluzole is a little bit, I never really know how to say it, riluzole or riluzole, but it's a pill. It's taken twice a day, and it has been shown in many clinical trials to prolong survival by a few months. So it's not it's not going to stop the progression of the disease. It's not going to reverse it. It's not going to cure ALS, but it does add a few months. And this is one of the most recent study looking at when it does add a few months, and it's really later in the disease. So I tell patients, if you want to take it, it's good to take it. The major side effects is some GI disturbance, and we have to monitor the liver enzyme because very rarely it can cause liver toxicity, but usually we see a trend, an increase in the liver enzyme, and we can usually stop the medication. Some patients don't want to take it or get side effects, and that's totally fine. It's not something that you have to do, but if it does add a few months. The other drug, edaravone Radicava, is a little bit more complicated. It needs a much longer discussion. One of the major reasons is because it's an infusion medication, so it's not just a pill that you can just do on your own. The infusion is usually every day for 10 days, every month, and because of that, you either have to go to an infusion center or you have to do it at home. You have to put a port and so forth. So it does require a lot of planning and energy. The other thing is that the data on it is, kind of controversial. And I see Mitsubishi Tanabe is there. They always come after I talk and tell me why you say that. But, I have to go by the science. The first study that was done was negative. It has a large number of patients. And then the second study that was done had a lower number of patients, was much more selected kind of criteria, restrictive criteria, and that showed at the end of 6 months of trial there was a change in the ALS-FRS score. And I can go in a lot of details in this, but again, this is an option that I tell the patient if they want it, good. If they don't want it, don't stress about it and don't worry about it. So now we go to supplements. If you Google supplements and ALS, you have over 29 million results. And I'm not going to talk about all these, but I think a good way to really address this is what the ALS Untangled group is doing. And anybody can access this website. ALS Untangled, Dr. Bedlag was here 3 years ago. Lance invited him, and he gave a great talk. He's a big fan of X-Files, and he always has flamboyant dress code. But really a great effort, because they really take medication, supplements, or drugs, or different ways of intervention, and they study it and see whether there is evidence or there's no evidence. And they classify the evidence whether we just don't know, so more research needs to be done, or there's evidence that we know that it doesn't work, or there's good plausibility that this actually could work. And a lot of time this will go on to go in clinical trials. There's many different papers written so far, and most ALS experts participated in some of these projects. And as you can see, you have the mechanism, the preclinical cases, trial, and risks. And I think the most important is always to look at the risks first, and F is bad. So you have over 5% of either significant side effects or death because of the intervention. And this one goes to stem cell transplant at the hospital San Jose Tecnológico de Monterrey. So don't go there. Other things, the U is unknown, and as you can see, the trials, most of them are unknown, so we just don't know. Some of them are, for For example, hyperbaric oxygen, there's been a trial and it's been negative. Antiretroviral, there's been a trial and back in New York, and that was negative. But there are more newer trials right now going on in antiretroviral, and I think it makes a little bit of sense. So all of these, you can actually access it for free and click and read the paper and make the conclusion that you like. But they also write a conclusion as well. Not only that, if you're interested in knowing more about certain compounds or certain medication, you can go and vote. And the medication that gets the highest vote usually has a priority for review and studying. So, and you can also suggest medication as well. So that being said, I'm not going to talk in details about every supplement. That will take us days. But overall, most of the supplements, multivitamins and so forth, unfortunately, don't have good evidence that they actually help. We studied vitamin D in the past, and that could have some benefit, but we really don't have any good proof. Vitamin E has been studied extensively. Again, no good proof that it actually does anything. Apart from people who are at risk of developing ALS, then there are some suggestions that if you take doses of vitamin E, it may delay the onset of the disease. Another common supplement that is taken is CoQ10, and it's a little bit expensive, but again, no good proof that it works, and so forth. Now, the reason why I'm talking about this in detail is because just this week I saw a patient who told me, I'm very excited for the symposium this Saturday. I hope you're going to talk about L-serine. I was like, no, I didn't have it, but now I have to put it. So this is a special request, and that's why it's there in a little bit more in detail. It actually has some interesting thoughts behind it. So L-serine is an amino acid which your body can actually make, and you can also buy it, and it's cheap and it's widely available. And the reason why there's a thought that it may work in ALS is because back in, was it in the 50s or 60s in the Guam Island Pacific, there was kind of a breakout of ALS plus Parkinsonism. And there was many theories of why this happened, but one of the things that got implicated was BMAA, which is found in the food that the indigents there used to eat. And L-serine can actually stop the toxicity of BMAA. So it was thought maybe we can do that in high dose, and that can stop the disease. One of the issues is that when you do autopsies on these patients, we don't find high dose of BMAA, although we know that BMAA is toxic to the motor neuron. So, and of course, L-serine has been reviewed by the ALS Untitled group, which concluded that it's unclear if it works, and there's really need for other trials. There's been one trial that looked at 15 grams twice a day, so kind of high dose, and they didn't find any significant toxicity. So it's probably safe, and there's an ongoing study right now, and see what that will go. So this is my just 2 slides on L-serine. And now diet is another big thing that is talked about in ALS, and we know that people who are malnourished, who are very thin, tend to do a little bit worse than people who have good weight and who are a little bit overweight. Unfortunately, we don't know what type of diet is good for ALS, so some people think high carbohydrates, some other people think ketogenic, but we don't really have any strong proof that one is better than the other. My thought on that is that, if you like something, just eat it. So I think that just eat whatever you like to eat, but at the same time, don't force yourself too much to kind of gain weight or something like that. Because a lot of people spend a lot of energy and time trying to gain weight, and I think you should just do what feels right to you. Exercise, hopefully, will be, thankfully, not hopefully, will be covered by Dr. Anshrud. And then we'll talk a little bit about ALS progress in clinical trials. Unfortunately, there's over 50 clinical trials that have been completed now with ALS, and we still don't have any cure. However, I think things are going to change soon. And one of the reasons is And that's, I think, I'm very happy that Dr. Harms is here because genetic therapy and modification of gene expression and RNA is real. It's no longer science fiction. Actually, in my clinic right now, I use 2 drugs in other diseases, not ALS, but in other genetic conditions that actually have shown to improve dramatically the outcome of patients. So this is real. The problem is that most ALS is not linked to genetic mutations yet. But about 15 to 20, and Dr. Harms can update us later, I think about 15 to 20% of patients with ALS will have some sort of genetic mutations. And when we have a genetic mutation, we have a target. And it makes sense to go and work on that target and try to fix the gene or tell it to stop expressing itself. And this way we can stop the disease from happening. So I think this is, to me, the most promising things going on right now. And that's why I put a lot of weight and importance on what Dr. Harms is studying. And as you can see, you know, for many years, the only gene that we had was SOD1. And then just in the last 5 years, there's over 30 genes that have been discovered. And I just heard last week you discovered a new gene, I think, or a couple of weeks ago. So it's just amazing how things are going so fast. So what is and I'm not going to dwell on this a lot because I think we have better experts who are going to talk about this. But this is very, very simple. Basically, you have a gene that is coding for a protein that's causing toxicity to the nerves. And what we do, we just send an antisense oligonucleotide that just attaches to the RNA and tell the gene to stop expressing itself, and the protein is not expressed. There is currently a trial in SOD1, and it's currently active and enrolling. And Providence will be a site for this study. So just last week, we diagnosed a new patient with SOD1, and we sent him to San Diego because they're doing the study already there. But once Providence starts, we're going to transfer back, and I think you can talk to the Providence team here who can talk more about this treatment. But I think this is very promising, and I do encourage every patient to, if they have genetic mutation, to participate in those trials. There's also a trial going on for C9orf72, which is much more common than SOD1. As Len said, there's over 100, and I have 5 minutes, I'm almost done, over 100 clinical trials going on in ALS at any time. So this is really good news. We've recently stopped enrolling for the Amylyx trial, which is run out of Mass General, and we're just going to wait to see what the results will show. We also recently stopped enrolling for the cytokinetics trial, which unfortunately was negative, but has some promising things, and I think the company will move forward to do more studies on that, and then we'll have updates for the copper with Dr. Beckman a little bit later. So I think the last question, and one of the most common questions, is stem cell treatment. And you know, stem cells sounds good. You know, sounds like something that could, I don't know, attractive, like science fiction almost. But if you think about it, what is stem cell? They take cells that kind of have the possibility to differentiate into different things, including motor neuron if you want, and put them back in the human body. There's 3 main ways to do it. One of the ways is to inject it in the blood. Unfortunately, when you inject the stem cell in the blood, they go to the lungs, they go to the kidney, and they stay there, and they don't go where we want them to go, which is the spinal cord and the brain, because that's where the motor neuron live. So that's not a very good strategy. Unfortunately, there are a lot of clinics everywhere that do that, but it probably doesn't work. The second way to do it is to inject directly these stem cells in the spinal cord or in the brain. However, you can imagine how many times you have to inject to cover the whole spinal cord. It's almost undoable. And the study that was being done is not continuing because I think of that major challenge to do it. And then the third way that I think is the most promising, and we have a phase 3 clinical trial, so very exciting. Phase 3 is when this is the last step of a clinical trial. If it's positive, then usually they would go to the FDA, and if it's negative, that tells us it's a good evidence that it won't work. And we do have a clinical trial, Phase III right now, where you inject the stem cell in the spinal fluid, and it would go not as good as when you inject them in the spinal cord, but spinal fluid be around the brain, around the spinal cord, and will go. And in general, the idea is not to produce a motor neuron that would go all the way to the muscle, because that will take years, but more to kind of create a healthier environment for the motor neuron to kind of slow down the progression of the disease. So slides courtesy of Dr. Goyal at UC Irvine, who's one of the sites that is running the trial, and we have a few patients who travel from here, from Portland to there to get the stem cell. So this is moving kind of fast for clinical trials. Phase II was rather promising. Data was published, and still data are actually being published even as we talk about that. And right now we are in the Phase III. So it thought that I'm going to go through those. So these are from the Phase II, show promising things going on there, and the growth factor and kind of protective factor around the nerves. And these are the sites: San Francisco, Cedars-Sinai, UC Irvine, and Mass General, UMass, and Mayo. These are the inclusion criteria if anybody's interested in this trial. Less than 60 years of age, breathing function more than 65%, less than 2 years of ALS, and kind of progressing, not people who are not progressing. So the good news is that they expect that in mid-2020 we'll have some results to tell us whether this actually works or not. And I think if it works, it will kind of quickly move to a clinical practice and commercialization. So this is, I think, my last slide. It's about what's going on right now at Portland and OHSU and Providence. We have the Orion, which is mainly a pill that you take. It makes the muscle a little bit contract, a little bit stronger, and the main outcome is the breathing muscle. There's biomarkers going on at Providence. There's deep phenotyping in patients with ALS at OHSU, genomic translation, or GTAC. This is at OHSU and led by Dr. Harms, who's probably going to talk a little bit about it. And as I mentioned, the very exciting SOD1 that is going to be happening at Providence soon. So you probably know this from last year. I ended up with the same kind of slide. This is really a very exciting slide to me because it really shows how things have changed over the years. For many years, there was like about 100 publications every year in ALS. However, look at the last few years. It's really, like just last year, over almost 1,700 papers published on ALS. So there's a lot of interest, there's a lot of research going on, and I think this gives us really hope and to continue to try, despite of all the failure we've had, to try to find something that works. And I think right now with the gene therapy, with the stem cell, I think we're really on the right track. So thank you very much for listening.
So our next speaker is a longtime supporter of the ALS Association and a longtime presenter at this conference. It's Dr. Joe Beckman, PhD. He's principal investigator and Burgess and Elizabeth Jameson Chair in Health Span Research at the Linus Pauling Institute. He's the Distinguished Professor, Department of Biochemistry and Biophysics at Oregon State University. A major focus of his career work has been investigating how oxidative stress, superoxide dismutase, and zinc are involved in ALS. He's been the leader in the translational research on a copper compound, copper ATSM, as well as other innovative therapies. And I like to share this personal story because I've known Dr. Beckman for many years, is that he does not just see ALS as a interesting puzzle to figure out. He really does see the human side of this disease. The first time I met Dr. Beckman, he actually brought a person living with ALS, drove him from Corvallis to Portland for an ALS support group himself, because he felt like that would be the most useful thing to help that person. And he's always made an effort to make sure his lab students and his graduate students participate in ALS events and participate in ALS walks, because he wants to make sure they know the human side of this disease. So please join me in welcoming Dr. Joe Beckman.
Thank you. I really appreciate being invited back. And in preparing for this, I went back and started to look at what I had said 10 or 7 years ago. And it gets a little frustrating sometimes of how slow the progress is, or you'd like to go faster. So what I'd like to do today is talk about this compound copper ATSM, which is showing some promise. And there's a lot of actually debate, or the right to try and so forth. I wanted to try to explain some of the reasons why drug development goes slow and some of the challenges with it. So it's being developed by a company called Collaborative Medicinal Developments, which is really only half a dozen people. It's a virtual pharmaceutical company. I'm a scientific advisor for them. I don't have any financial connections, and I receive no funding from them. But I like to say I'm their cheerleader in trying to say these are things that we need to move forward. I also have another company that develops mass spec, because we've actually had to invent a lot of different technologies for the work we're doing. And this mass spectrometry technology is helping us understand what's happening in ALS, but it actually has a lot of other implications. It's basically moving beyond DNA and looking at the tangles and all the other things that are going bad inside of the brain. And I have a personal connection that just started or came up with a friend who was in the military who developed ALS. So I'm going to start with this picture of an aluminum canoe in my backyard that my wife keeps wanting to give away. So I got out of the Army in 1979, and I had been in Korea. And there was this ad that was kind of amazing. You could buy a canoe for 5 boxes of Uncle Ben's rice and $150. So I got the 5 boxes, I sent it in, I bought a canoe, and I called up my friend who I had known in the Army, and that was Tom Bradshaw. And he was a West Pointer, so that made me a little nervous at first when I met him in Korea. Pretty straight-laced guy, but he was a true gentleman. And I think there's a lot to really nice people end up getting ALS. It's completely unjustified. So Tom and I put the canoe on top of a car. We took 2 weeks off, and we went canoeing throughout Maine. So I lost touch with him, actually, after a few years, and I was landing in Rome last summer, and I got an email from Tom, and I thought, this is really great. But I opened it up, and he said, I just learned I've got ALS, and I will find out that you're working on it. So it's one of those, there actually is a large community of veterans, too, that are developing the disease. So the Ice Bucket Challenge happened in 2014, and it's been about 5 years, and you may wonder about what happened with it. Why after 5 years aren't we there? Well, I've actually been working on the disease for 25 years, and I feel like I understand in some ways less about the disease than what I thought I knew a few years ago. And I just want to briefly mention, so the photo on the right is the president of OSU, Ed Ray, and he's about to have ice water dumped on his head. And the woman in the middle, Pam Bilby, did her PhD with me. And I want you to remember that, because at the end, she's actually helped develop another drug that I think's got a lot of potential for treating the disease. And it's one of these things of looking at a lot of different places is really important for the disease. And I wanted to use this analogy last year. I've been studying swarm behavior for optimization. But it's basically the idea of, if you have flocks of fish or birds floating around and they're hunting for insects, that they go their own ways and start to search, but if one starts to have success, the other bird's kind of watching and doing its own thing, but it's seeing, how's he doing? And then pretty soon they can start to swarm in, okay? And basically, swarm optimization works pretty well, and it's something that the pharmaceutical industry is really good at. Once you have a pretty good lead, you can bring a lot of people, a lot of resources to it. You find a rich area, and you can improve that. Things like the ALS money basically help you. A lot of people search out in space where you don't see anybody yet and find those first insects that are tasty to bring them in, okay? And that's one of the key things about research, and I'll come back to this. there's a lot of things that are really promising, but there's still a lot of space to search. And it's important to keep doing that searching. And you heard about collaborations, and one of the great things for me is basically I've met a lot of friends around the world. And a number of these, this is the one point in South America where you can stand at that particular spot and you can watch the sun rise and set over the Atlantic. So it's in the country of Uruguay, and the arrow points to that spot where we were standing. But Alvaro Estevez is now with me at Oregon State University, and we've been collaborating for 25 years. And Luis Barbedo is also, we've been working together. And there's another drug called mestinibid that came from Luis's work and his graduate student, now Dr. Emiliano Trias. So that collaboration with South America has been great. And the other collaboration we had is in Australia, which I really like this photograph when I was looking for an upside-down map of Australia. But you can see here the cat's head and then the dog's head. So there's a group in Melbourne that discovered copper ATSM in treating ALS. And I had a graduate student who went down to Melbourne and was working there, and he brought it back to my lab. And so this is a very important day for us that Blaine Roberts, who's in the back, and his wife Ann, and a graduate student are working with our people in the basement of the ALS building, Agricultural Life Sciences building at Oregon State. But I had to have my lab there. And that's when we realized that we really needed to try copper ATSM in a new model of ALS. So in 2012, I gave a talk here, and I was able to show that the first results that a new drug actually kept these mice alive for a year. And that was a pretty phenomenal result, which, because the SOD model of mice, which I'll explain a bit more about, is really harsh. It's really hard to treat these mice. And this is the compound, and it's copper ATSM, okay? And basically, the orange ball in the middle is an atom of copper, and it's surrounded by a very flat planar molecule that's hydrophobic, and it can carry copper into the brain, okay? And it was used as a PET imaging agent to take radioactive copper to visualize tumors. And so we knew in humans it gets into the brain immediately. Crossing the blood-brain barrier is not a problem. It happens in minutes. Well, a lot of the work that I'll talk about has led to a Phase I trial that was done in Australia. And here's some of the results that are coming out from it. A Phase I trial is basically just to determine safety. Is the drug safe? But the nice thing about Phase I studies, it's usually done in healthy male volunteers, college students who will do anything for a little extra money. But in this case, you could take the drug directly to patients. And one of the things that the Australian review committee said you could do is the idea was to treat for just 28 days, but they said, well, if it's tolerated well, we want you to extend it to 6 months. So in fact, this Phase I study actually gave a lot more data about long-term treatment. So there are 6 patients that were treated per dose, and they started at a very low dose of 3 milligrams per day. And they're at up to they actually are beyond 72 milligrams per day. But that's going to be the dose going forward. And so there are a lot of blood draws and a lot of safety. Different things were done to test the efficacy. But it's important to know that this drug actually can't tell you if the drug works, okay? It's just a very small study to see is the drug safe. And to explain what's going on here, I need to explain a little bit about copper. So each of us has about 100 milligrams of copper in our body. And we breathe in oxygen, and essentially every molecule of oxygen we breathe touches an atom of copper in order for the body to use it, okay? So it's critical. It's like an electrical conductor that's needed for us to be able to make energy and to defend against oxygen. Now, 100 milligrams is a piece of copper wire that's about that long. And the body regulates copper very carefully because if you make it twice as long, that's a lethal dose. It causes all sorts of problems to the liver and to the kidney. And to evaluate the safety of copper ATSM when you're doing 72 milligrams a day, you're giving every day about 30 milligrams of copper, okay? And what we're doing is wrapping it into a molecule that can carry it into the brain and into the liver very effectively. So when we presented this to pharmaceutical companies and to study sessions and so forth, they say, you're crazy. You're going to kill people with copper toxicity. And it's a very reasonable expectation, a very scary thing that you have to worry about. The good news is that it holds on to copper so tightly that most of it is secreted and it releases copper really to places that's needed. So it turns out the safety actually works. And now we have a dose, or as it's been identified, to move on to phase 2 studies where you randomize the drug and start to see what it can do. There we go. So last December, the first results from the phase 1 trial were talked about at a motor neuron meeting in Scotland. And it mentions here that there's some very promising studies, and this has been picked up and created a lot of enthusiasm. So this is just the rate of progression. So what happened at the 3 lowest doses is there was almost no effect on the disease progression. But at 72 milligrams, the progression is less than half for other patients. And this is still 1/10 of the dose we had to give to mice to have efficacy, so it was about where you would expect to see a result. But keep in mind, that's only 6 patients, and they're not randomized. Okay, so you really can't say that this is going to generalize or be a treatment. The second is that the forced vital capacity measurements actually improved. And that actually, no one's really seen that before in trials. So it's created a lot of excitement about physicians, and it's really helping accelerate the interest in moving this to phase 2 trials, that in several of the patients, their respiratory function actually increased at the higher doses. They also did a study in Parkinson's disease, and the drug is actually showing even better effects in the Parkinson's patients. So this is a functional scale where going down is doing better. So the drug is doing a little bit more than you might anticipate based on the animal studies. So in summary, what's been learned from the phase 1 studies? It's relatively safe, but there are some patients that had to withdraw from the studies. The copper ATSM is very tough to formulate, okay? So patients were having to take 24 large pills. And for patients that have difficult swallowing, taking big pills is a problem. So the other thing is that the absorption is variable. So they have to reformulate the disease, and they have to go through another phase 1 study to prove similar results are occurring. The good thing is that there's low doses have no observable effect on the disease progression. And right now, you can't say that this drug actually works, okay? Even though it looks promising, there's all sorts of statistical reasons why you can't say that this is truly working. And so now they're planning on the phase 2 trials, which is basically scaling up and starting to look for efficacy. And one of the things to keep in mind is what will it cost, okay? So, there's a joke that is, how do you make a small fortune in the pharmaceutical industry? And that is, start with a large fortune. So just to make enough drug for the phase 2 trials, I can't really talk about details, but you can put it in the range of $1 to $10 million, okay? Just to treat the next 200 patients. And then when you go to the phase 2 trials to actually do the clinical work, it's in the range of $20 to $80 million, okay? Depending where you do it, how many people you enroll. So it's putting a big bet on this right now as to seeing what happens. And it's understanding that there's a lot of different processes that play, a lot of different opinions that you have to come together and try to rationalize and get a lot of people to agree. And that's one of the reasons why it takes so many years to get this moving. And then really, who's going to step up and say, I'm going to commit those kinds of resources to make it happen? So I want to step back and say I am a basic scientist, and I really have started to study this disease because of the interest. And it goes back, the first mutation that was discovered was in 1993, and it was SOD1. So it's a protein, and we've heard about different genes and how the DNA is moved. I actually have studied SOD since the early 80s, actually late 70s. And Erwin Friedovich, the man on the top who discovered it, was on my PhD committee. And we were working on this for a lot of reasons. We actually turned it into a drug to treat stroke. And we discovered a side reaction that could be potentially toxic. And when the SOD mutations were uncovered, that's when we started to work on SOD and trying to understand the physical basis of it. But one of the key things here is at the center of this is a copper atom. Okay, and that's the reason why copper ATSM, why we first went to investigate it, because we found that in animal models, copper was missing from a fair amount of the protein. And it's a real dilemma. We've heard that 2 to 3% of patients have mutations, but within that one gene, there's over 180 different mutations that have been discovered. And the crazy thing is, moving one carbon atom in this protein is enough to cause the protein to become toxic, okay? And yet you have a lot of this protein in every cell in your body at birth, and you can be 80 years old before the disease starts, okay? So there are a lot of puzzles to figure out about what's going on and what's causing the disease. And we've approached this in many different ways. I'm a biochemist, we produce the protein, we've studied it to death as many other people have. We work with cultured cells, and we work with motor neurons, which you can purify. And we can study a process that causes these cells to die, and we can establish that mutant SOD will cause that problem, or it can also replicate this in a test tube. And we also work on the cells that surround motor neurons. We tend to think of the brain and the spinal cord as being neurons, but you have 10 times as many glial cells around it. And these are support cells, and these get changed. And so the yellow here is showing one process that's showing other cells that are causing damage. And it's really key because that's probably what's driving the progression of the disease. We lose motor neurons throughout life, but what causes one set of motor neurons after another to progressively die? And what we're looking for is a way to stop that progression, because then you could turn this into post-polio syndrome. And out of the 100 genes that yet to need to be defined, a lot of them are going to affect that progression process. And so one of the things, there are all those mutations that have been discovered, but the amazing thing is that copper zinc SOD is the one mutation where you can take that piece of DNA from a human and you can put it in a mouse or a zebrafish or a rat or a pig or fruit flies, and they get motor neuron disease. If you do that with the other mutations, the other 50, you really don't get a very good model of motor neuron disease. And that's been one of the problems, is trying to figure out what do all these other things do that's related to motor neuron disease. It also turns out that dogs have a spontaneous mutation in SOD, and so many dog breeds are susceptible to developing a form of ALS. So it's not just humans that have this disease. And this ALS phenotype, this motor neuron degeneration that happens in mice and rats, is really robust. But the model's actually fallen out of favor. And part of it is shown here. So the mice will develop the disease. They'll look fine till about 100 days. And within 30 days, they develop the phenotype of this. So someone talked about they didn't harm mice. I'm afraid a lot of mice in my lab develop ALS. And so this was one treatment that was tried that's a common antibiotic that's thought to affect the glial cells or the microglia. And they were excited about this 10% extension of life. The NIH put $20 million into doing a clinical trial on it, and it failed. But if you look at this, the mice all died at the same age, okay? So there's $1 million prize if you could extend the life by 25%. And no one could do it for a number of years. So we were really excited when we had copper ATSM and we hit that 25% extension of life. But there was something else that we were studying. Well, I need to step back for a minute. So this SOD mouse also became kind of a poster child for the importance for rigor in science. So this is a graph of studies that were published showing a protective effect of some treatment that's in the light green, blue, green bars. And then if the same drugs were tried by the ALS therapy development in Boston, they had zero effect. That's shown in the black bars. And so that kind of contributes to this idea this model yields random results. So I mentioned that we worked closely with people in Australia, and Peter Crouch is an important collaborator. He and I did the same study at 2 different doses, and he put this graph together that showed basically as we increase doses from 2 different places, you get a linear response. I was really excited 2 years ago, maybe it's 3 now. The ALS DDI repeated the study, and they got a protective effect, and a pharmaceutical company, Teva, actually repeated this at 2 doses. So multiple people can repeat the study, and that's actually never been done before in ALS. And we were actually studying something else about what makes the disease worse. And the copper ATSM, in this case, actually was extremely protective. So we had mice, this is what I presented 6 years ago. If we were able to take mice out to a year, we repeated the study in a more rigorous fashion with more animals. And the animals went out to 2 years, okay? So the curious thing is when I present this to the ALS community, they're amazed at the 25% extension of life. Nobody believes this, okay? And no one's ever repeated it yet. We have, but so it was too good of a result to be true. It actually took us 2 years to publish the result too. Now, people are saying you should only try this drug in familial SOD patients. The Australian studies are actually done in sporadic and familial patients, and the effects are protective in both. I've been working with Chris Shaw, who's up in Vancouver, Canada. He's been studying the Guam form of ALS, and he makes a very strong case that it's not that toxin BMAA, but rather there's a cholesterol molecule with a sugar attached to it that's causing the problems. It's very hydrophobic. So he's been studying this for quite a long time, and we collaborated on this experiment. It was just published last week that the copper ATSM was able to protect these mice from this toxicity. People tend not to remember, but the brain is actually people think of the liver as making cholesterol, but 25% of the synthesis of cholesterol is in the brain. So there's a whole chemistry of lipids that's really critical in the brain. Now, why would this happen? It's not an SOD model. And so there are organelles inside the body that produce energy, and copper ATSM is also doing things to protect the mitochondria. And I'll come back to that in a minute. So we've spent a lot of time of developing different derivatives of copper ATSM and testing to see how they will work and trying to find ways. But what we're excited about is this is finally moving forward into patients, and it looks like we'll find out in another year or 2 whether it's actually going to work generally. But I'm very excited that it's actually working in a broader range of diseases. And there will be an abstract presented at the next motor neuron meeting in Perth, Australia, that'll show it works actually on another major mutation that causes ALS. So we've had, I really set out, I'm a PhD working on pine trees that started to study ALS. But because of understanding the mechanisms, we've actually had a lot of luck of discovering new drugs that have a lot of potential. So there's copper ATSM. Mestinibid is a drug that inhibits glial proliferation. It works on mast cells. And so with Luis, we've actually managed to get enough studies that's gone into phase 3 trials. We've done all the preclinical work for that. We think Riluzole works by a completely different mechanism that other people will do, and we think if you put it in discontinuously, give it for a few days and stop and start again, it will work better. It definitely does in mice and in cultured cells. The reviewers so far are saying that it's unethical to try it in patients, but we'll keep hammering on them. And then we've heard about Darivon or Radicava, and there's another trial using urate. That actually works on the SOD-based mechanism, so we're studying what that is. But I want to show you that we've been working on something else as well. So the first part is that when you're doing basic research, you're really hitting your head against the wall. ALS is a really tough problem with many layers. And you can see I like water themes a lot, so I like this slide of basically we're going to try things and we will wreck on the rocks fairly often. But you still need to do basic research. So this is Blossom Bar on the Rogue River, and it looks impossible to get through. But in fact, there's a way that you can go through it. But this is what you're doing with basic research, is you're exploring lots of channels, and you're going to hit a lot of rocks. You're going to fail 99% of the time. But there is a way through it when you figure out how to go to the left and go to the right and back paddle here. And that's how you're going to find a path to do what looks like it's absolutely impossible. So the last one, I want to show what we've been doing with the money that we got from the Ice Bucket Challenge that Lance arranged for us for a few years ago. So the first diagram shows a cell, and inside of it is a mitochondria, which is often called the engine. And my collaborator, Tori Hagen, was showing that there's a small molecule that gets depleted and really damages mitochondria. It's a natural antioxidant. And it's a completely different compartment in the mitochondria. So we had developed a lot of synthetic chemistry capabilities. So we were able to attach a molecule to it that would drag it through the membrane and accumulate in the mitochondria. Now, it sounds crazy to do this because it's actually bigger than anyone had done before. And getting it to work in cells would be amazing. But what we've seen in the past years, we actually gave this to a mouse model of Alzheimer's disease. And so this is showing a picture of mitochondria. So this is a healthy mouse, and these are mitochondria in an electron microscope. In this Alzheimer's model, the mitochondria become vacuolized, they blow up. And when I saw this picture, I was amazed because it's actually what you see in the SOD mouse models and some of the other models, a lot of mitochondrial damage. And just injecting that compound every other day into the belly, it actually got into the brain. And so that's a control for that. And you can see the mitochondria over here are not looking like this, but more like the controls. There's actually, you can show lots of other functional data that it's actually working. So we're really excited about this molecule and trying to get it into down this path to clinical trials. And so hopefully in 10 years we'll have it there or faster. So I'd like to stop and thank you all for your attention.
Thank you, Dr. Ansrud. That's very informative. I look forward to a lot of questions, and we really appreciate some of the very practical, hands-on demonstration, and thank you for Dr. Karam for being a guinea pig today. Our next presenter, we're thrilled to have him travel from the East Coast to join us today, although he'll share from you his personal experience that this is kind of a homecoming for him. Dr. Matt Harms is an assistant professor of neurology at Columbia University in New York City. He has his medical doctorate from University of California, San Francisco. He completed his neuromuscular medicine and clinical neurophysiology fellowships at Washington University in St. Louis. He's past director of the Washington University Neuromuscular Genetics Program. At Columbia, he's continued his work harnessing emerging genetic technologies to understand the cause of inherited neuromuscular disease, including ALS. He leads an international multi-site team using whole genome sequences in an effort to bring precision medicine to ALS. So join me in welcoming Dr. Matt Harms.
Thank you. Yes. So Lance mentioned that this is a bit of a homecoming. I actually grew up in Eugene, so although I'm coming to you today from Columbia, I actually got my start here in Oregon, and just felt my blood pressure lower as I got off the plane and could smell the evergreens although that might have been the lack of exhaust fumes from New York City taxi cabs. I'm not exactly sure. So today I want to talk about the genomics of ALS. We used to call it the genetics, but that's because we could only study a limited portion of the genome. And now that we can do the entire thing for relatively cheaply, we're starting to call it the genomics of ALS because we're starting to find out about regions of the genome implicated in the disease that we didn't know about previously. And I want to talk today about how we're using that to guide trials and then to generate targets for therapeutic development. So we already heard some of what ALS is, and I just want to remind everyone that it is a complex disorder. It's called a motor neuron disease, but now we're learning that, in fact, other neurons, including those in the frontal executive circuits at the front of the brain, sometimes those in the deep centers of the brain where the smoothness of movement is controlled, and then even some patients have involvement of what are called the dorsal ganglia, which control sensation. So although the motor neurons are the most deteriorated and the most affected by the disease, other neurons are involved. And within a given patient, there can be any number of combinations of these different regions that are involved, which means that in the clinic, every patient looks different one to another and makes it a challenging disease to study. So who is it that gets ALS? Unfortunately, many of you in the room know firsthand exactly who that is because you may be among them or your loved ones may be. But it turns out that actually this is what I would call a not-so-rare disease. So 1 in 3 per 100,000 will develop the disease in a given year. And that means that living around the world, there are close to 300,000 people with it. In the United States, that translates into about 30,000, or here in the Oregon, Washington, and Idaho region, about 750. So it's not that uncommon. We do know that there are some groups of individuals who are at increased risk. So males have a slightly increased risk. Elite athletes you heard about earlier, those who have served in the military, whether they've seen combat or not. And then those of European ancestry compared to African or Asian ancestry are also at increased risk. To put that in a little bit of context, that means for me, my lifetime risk of dying of ALS is actually 1 in 372. And to put that in reference, it's a little less likely than me dying in gun violence, which is 1 in 280. And a little bit less likely, or more likely, than being struck by a car and dying as a pedestrian. So it's not that uncommon, although I know sometimes it feels like you're all alone in suffering with this disease. So one of the strongest risk factors, of course, for developing ALS is having a strong family history. So when someone comes to my clinic, there's somewhere between a 7 and 10% chance that someone in their family will have had the disease at some point. And if you have a strong family history of ALS, your chance of getting the disease is actually about 1 in 3. And in some families, it's more like 50-50. But overall, it's about 1 in 3. And it's actually from studying those individuals with a very strong family history that we've learned the most about ALS, what causes the disease, by studying the genes that underpin this family history. You saw a very similar slide from Dr. Karam's talk illustrating the gene discovery history in ALS. So 1993, just as I was completing my high school time at North Eugene High School, was when SOD1, the very first gene, was discovered for ALS. And there's been slow and steady progress in the next decade. The human genome was actually published in 2000, and that gave a tremendous boost in the ability to discover genes, made it faster and simpler. But really, it wasn't until 2009 when a new gene analysis technique called next-generation short-read sequencing came online that the real pace picked up. And you can see that basically since then, it's just skyrocketed the number of genes that we know. And up here in this upper region, I'm happy to report that many of the projects were actually funded directly by the ALS Ice Bucket Challenge, something that many of you participated in and that got people very excited about helping out in ALS. So how do gene mutations work? So I thought I would just go through this at a very basic level in case you've forgotten some of this from high school biology. So the central dogma states that the DNA contains the instructions for proteins. And a copy of the DNA first gets turned into a set of code called the RNA, and then the RNA itself goes on to the protein. Because you inherited one chromosome from mom and one from dad, you have two copies of the CHOMP gene, which I've illustrated here. and 2 copies of the linker gene. Of course, you actually have 19,000 genes, but we'll just focus on these 2 examples. And so what first happens is a process called transcription is utilized, where the CHOMP DNA set of instructions is copied into the RNA set of instructions. Both both chromosomes will be utilized, so you'll have a copy that came from mom and a copy that came from dad. And then both of those, if they're normal, will get translated into a protein. And so here, the CHOMP protein looks a little bit like Pac-Man, because its job is actually to cut whatever this happens to be in half and leave it in pieces. So that's its normal function. Some cells will use the CHOMP protein, others won't. It kind of depends on what type of a cell you are, which of the different genes you're going to use. And so you can imagine another gene, the linker gene, gets used in some other cell types. And so similarly, it gets transcribed into RNA. And then that gets used to manufacture the protein. And in this case, it's the linker protein because it pulls these 2 other proteins together so that they're close enough to do the job that they have to cooperate to accomplish. So how does a mutation mess this up? So if we now take a look at the CHOMP and linker proteins and introduce an abnormality in the DNA, that abnormality in the DNA will now show up as an abnormal code in the RNA. And then when the translation occurs, the protein is defective. And so now the mouth of the CHOMP protein isn't close enough to actually cleave this rod. And so uncleaved, it just kind of starts to accumulate, and the clumps of the rod basically get in the way of the cell's function. Similarly, for the linker, if there's a mutation located there, this copy of the RNA will be abnormal, and it may lead to an abnormally globular shape. And so although this protein can now grab onto the red one, it can't bring the blue one close enough for them to do their cooperative job. So that's how a gene mutation leads to malfunctioning proteins. And then if that protein is important enough, it goes on to lead to problems in the neurons and cell death. So all of those genes that I showed you on that graph have functions. And so if you start to look at the functions of all the genes we've discovered in ALS, you can start to see some themes that tell you which pathways are really important for keeping motor neurons alive and which pathways are going wrong that lead to ALS. And so if you put them on lists, you can start to see that a process called protein homeostasis is really important, a process called cytoskeletal and axonal transport, where things that are manufactured up here in the cell body have to get all the way out here to the very end where the muscle's located. We now know from these types of analyses that metabolizing the RNA, that middle component that I showed you previously, and then shipping neurotransmitters and other things via endosomes are also important. So one of the things that we've learned from uncovering the genetics of ALS is not just who has carrying the gene, although that's very important, but we've also learned kind of which pathways are super important for keeping motor neurons alive. So how frequent are mutations in these genes? So we did a study just as those next generation sequencing techniques were being released, which allowed us to look at a large number of patients for the very first time. So in the WashU Neuromuscular Genetics Project that was mentioned earlier, we had 42 individuals who had a very strong family history and 350 people who were so-called sporadic ALS. No one in the family had ever had the disease. And we were able to, because of the cost of sequencing coming down, analyze 17 ALS genes across both of these groups. Previous to next-generation sequencing, it would have cost us about $500,000 to do this study. With the falling costs from next-generation sequencing, we were able to do it for about $25,000. So there was a huge savings. And if I redid this study today, it would probably only cost me $2,500. So the cost of sequencing has dramatically fallen. And what we learned from this study is that we could find a causative gene mutation in the C9, SOD, TDP, these other genes, in about 60% of families. And in sporadic ALS, we were surprised to find those exact same genes and those exact same mutations in 9% of people with sporadic disease. So completely unsuspecting that they were carrying a gene mutation. But that means that there's, I guess I should also point out that we've now gone from analyzing 400 patients to analyzing 5,400, and the numbers still hold true. We're learning new, interesting things by this much larger analysis, but this illustrates just how broadly sequencing can happen now because of the costs coming down. So that still means that in sporadic ALS, we don't find an explanation in 91%. And if you look over in the familial cases, even to this day, we still don't find an answer in about a quarter of individuals. So that means that there must be other genes out there or other things going on. So what else is going on in sporadic ALS? So I've illustrated for you those genes that cause ALS, where one bad mutation is enough to trigger the disease. And so that's one thing that could be happening in that 9 or 10% of sporadic disease. But it's also possible that you could have 3 minor genetic problems that kind of all pile on one to another to trigger degeneration. You could also envision, though we haven't discovered one yet, a massive environmental exposure that could trigger neuron degeneration. Although viruses, some types of viruses are implicated in ALS, the West Nile virus, as soon as you get infected, it kills off motor nerve cells. So you could envision an environmental exposure that was strong enough to cause ALS all by itself, but we don't know of one yet. I want to emphasize that. But you could also envision that there would be multiple smaller environmental exposures. So maybe being an elite athlete and in the military and a male and exposed to X, Y, or Z could all add together to cause disease. But probably in sporadic disease, it's a series of genetic problems plus a series of environmental exposures that eventually go on to cause the motor neuron to degenerate. And the technologies that we have today have allowed us to make lots of progress in identifying one major genetic problem. And we're just now on the cusp of being able to look at multiple genetic problems and this combination. So another way to kind of visualize how this is probably happening is that if this scale represents the health of your motor neurons, and a tilt to the right under the insults category leads to motor neuron degeneration, if you have a really big bad mutation, like the A4B mutation in the SOD1 gene, that is a massive insult. And although your body rallies and tries to compensate by expressing other genes, trying to turn down the SOD1 gene, it's just not enough and could never be enough. And so everyone with the A4B mutation, the balance tilts and they develop the disease. You could also then envision smaller effect mutations. So this would be another mutation in the SOD1 gene called the I113T. And the compensations in about 50% of patients are sufficient to prevent the disease from ever occurring. Yet we know that being male is a risk factor. So now if you add in just having only one X chromosome instead of 2 because you're male, that's enough to tilt. And we know that the SOD1 mutation I113T manifests more frequently in men than in women who carry the disease. So now you can also envision taking that one step further to these smaller effect mutations, and these wouldn't necessarily have to be in the same gene. And so you could have 3, 4, 5, who knows, we don't know what the upper limit is, little tiny mutations that minorly affect 3 or 4 different proteins. And those compensations could be quite adequate. And so many people could be walking around with these same risk factors. And then you could add in cigarette smoking or some other little feather of a risk factor that adds in and triggers the disease. So what we think of ALS from a genetic perspective is that it's a complex disease. And so there are situations where one gene mutation is sufficient to cause the disease, and at the other end of the spectrum, a pile-on effect or synergistic effect from lots of different minor genetic problems, plus some environmental risk factors or lifestyle factors, and that most people are somewhere on this spectrum. And if they're at this end, we call it monogenic, or one gene, and there's a single cause. If it's at the other end, we call it multifactorial. And then really we're going to recognize these individuals as familial ALS because those big bad gene mutations are really strongly active. And then this is what we're calling sporadic ALS at the other end. But it's a spectrum and a continuum, and they're all genetically influenced. So while you may have sporadic disease, there are still genetic contributions that are playing an important role in your disease, though they may not be these big bad mutations that you're likely to pass on. So I'll go back to this ALS discovery timeline because there are now, I would say, 20 to 24 causative gene mutations. There are actually 100 claimed. So there are a lot of genes that aren't actually ALS genes that have been said to be. But that raises the question, how many undiscovered ALS genes are there? And there are some complicated statistical models that actually come from ecological estimates of species in forests where you can't go out and count all of them. And it suggests that there are 100 yet to go for us to have identified all of them. So that begs the question, why continue the gene hunt, which is what I do? So there's job security, so I may be a little bit biased, but I think that there are some good scientific reasons. So the first of these is to provide answers to some key questions that are definitely pinging around inside your heads, which is, why me and not someone else? And could this happen to those I love? Could I pass on genetic influences that make them more likely to develop the disease? And that's a very important question, and one that I answer every day in clinic as we do these genetic tests. More importantly, I think kind of to ALS in general, is that we want to be able to empower what we call precision medicine. So the idea for precision medicine is that we're giving the right drug to the right patient at the right dose at the right time. And so we want to be able to use genetics to help power the next generation of clinical trials, to identify responder groups for drugs that we already have. It could be the reason why Riluzole doesn't look like it helps that much is because we've given it to everyone, and that there's a subset who do really, really well on it and a subset who do really poorly, and we could be able to distinguish that. We also want to be able to use genetics to guide the best choices among drugs once we have a whole host of ones to choose from. And then I think most importantly, we really want to identify new targets, things that might be easily druggable and fixable from a therapeutic perspective. And that could include the individual proteins that we learn about and then important cellular pathways. So how do we do that? So Shafiq already alluded to the fact that we've now had 60, 70 randomized clinical trials that have led to 2 approvals. And what should we have discovered if we were like the other diseases that the pharmaceutical industry has targeted? So typically 10% of drugs in trial make it to FDA approval. So by my calculation, that means that we should have approximately 6 by now, and we only have 2. So what's been the problem? Well, one of the problems goes back to this snapshot here, and that's that we take all comers in our ALS clinical trials. If you meet the clinical diagnostic criteria and in the inclusion and exclusion criteria, you make it into the clinical trial, and that basically assumes that ALS is one disease, that everybody coming to the clinic, everybody joining the trial, has the exact same underlying mechanism, and I think the genetics is telling us that that's not the case. So some of the problems with the ALS clinical trials that have happened in the past not proving that the drugs work, some people call them failed trials. I like to call them negative trials because I think we did learn from them, and failure implies that it was just a waste of our time, and that's far from the truth. So I think we learned that there are some drug-specific issues in the way we used to do studies. Number one, we didn't realize that most of the drugs we were trying didn't get across the blood-brain barrier and actually get to the neurons. They were in the bloodstream, they were in the liver, but didn't make it to the brain. So obviously had 0 chance of saving neurons. We also learned a lot about study design issues. We were choosing too small a cohort. We were doing the studies for too short a period of time, and we were shooting for the moon and anticipating a much larger effect size than I think we've learned to expect. But I think the major reason is because we've been ignoring the big difference in underlying causes amongst the different patients that are coming to clinical trials, and that in the past we've been limited in how we can look for targets for drug development. We really only had the SOD1 mouse model, and we really had no way to look back in human tissues to find out whether the genes we were studying were important in the disease or not. So we've made a lot of progress. We now think about this routinely at the very, very beginning of thinking about a drug for clinical trials. The very first thing that gets tested is, can it cross the blood-brain barrier? How long does it reside in the system? Things like that. We now have very sophisticated design trials that morph from phase 1 into phase 2 and take some of these issues into consideration. And so what I'll talk about the rest of the time is kind of how genetics is playing a big role in these last 2 components. So again, here's the typical ALS clinical trial. They meet the clinical diagnostic criteria for ALS and come into the trial. But we really know that if we could look at the underlying biological cause, we would start to see differences in different subgroups of patients. And so basically, if we could figure out a way to find the individuals with the same underlying causes and parse them into these different groups, then we could perform genetic stratification. This would mean that the participants in a given clinical trial would be more similar one to another and would make the trial easier to perform. It means that we could then use smaller numbers in the clinical trial and potentially even lower the number of people receiving placebo. This would translate into much cheaper trials that could happen more quickly and give us a better chance to see the effect, especially if we get to the point where we're able to pair one of these individual subgroups to the type of drug mechanism that's being proposed. So if you knew that this blue group had a problem with pathway X, and the drug you were developing seemed to target pathway X, you could pair up just those individuals with that particular drug study. So that's what we're moving towards in the future by doing these genetic investigations, so that we could take this cohort of ALS patients that all look the same on the outside and figure out who they are and their underlying biological cause. And then figure out, is there a group that's basically neuroinflammation or cellular transport, or what are the genetics and the features of these different groups telling us is going wrong, so that then we would be able to target the right drug that affects each of those different pathways at the right time. And so that's the future, the promise of precision medicine for ALS. And I think at the moment, we're already doing this in some of the strongly genetic subgroups. We would also anticipate that we would find people who didn't fit into these nice, neat categories. And these are just hypothetical categories, by the way. This isn't known yet. So this individual might not have the features of any single group, but might look a little bit like these two. And then we would say, well, let's just try kind of a combination drug where we give them these two and see if that works better than one by itself. So already this is happening for strongly genetic subtypes. Dr. Karam already told you about the anti-SOD1 antisense oligonucleotide. At the American Academy of Neurology meeting, the initial results were released, and in fact, the antisense oligonucleotide does its job. It gets across the blood-brain barrier, and it destroys the SOD1 protein. So that's very successful. And not only that, especially in the individuals with that A4V mutation, that big giant weight that sat down on the scale and tipped it all by itself, the rate of progression was arrested in some patients and slowed dramatically in others, which is not ever heard of in that particular mutation. So this is extremely exciting. And a similar molecule for a different gene, the C9orf72 gene, is also underway. And I needed to update this. I don't know where your nearby sites are, but there are some here. San Diego. Okay, great. All right. So going back to this slide, kind of showing the different pathways that are involved, eventually we'll probably have antisense oligos or similar types of drugs that target many of these different genetic mutations. And I would also argue that the more genes we discover are going to teach us what neurons need for health. They're going to show us some unsuspected pathways that aren't currently on this list. And once we have 15 pathways on the list, then we're going to have the opposite problem, and we're going to need to know which pathways are the most key ones for us to target, because we don't have enough resources to basically design drugs for all of them. Which ones should we zero in on and focus our energies on? So how are we going about identifying more genes? How can we get those 100 that are still hiding out there? And so that's what I'll talk about basically for the rest of my talk. So mainly the way we're doing this is by a fancy statistical test called burden testing. And I promise all of you can understand exactly how this happens. Okay? So if you have a group of controls and you sequence their whole genome, and here I'm just showing one part of one gene, and then you ask in these controls, people who don't have ALS, you ask the computer to show you all of the regions that are different from what we expected from what we call the reference genome, the normal genome. And then you see that in each of these different locations where the stars and lightning bolts are located, there are differences in each of these individuals. Then you ask the computer to predict which of those changes are likely to disrupt the protein, to prevent the linker from bringing those two things together or prevent Chomper from cutting the rod, and those that are predicted to do nothing to the protein, to not affect it at all. And so you ask it to classify each of the different changes. And then you do the same thing for a large number of cases over there on the left-hand side and ask the computer to do the same thing. And now you basically compare. Are there more deleterious mutations in the patients who have ALS compared to the people who don't in any genes across the genome? So no one knew whether this would work back in 2015 when we did our first study. And so we assembled 3,000 ALS cases and 6,000 controls using this big consortium. And using generous funding from Biogen IDEC, at that time, about $5 million, we sequenced what's called the exome. So that's the region of the genome that makes proteins. It's about 2% of the DNA in our bodies. And what we were able to do is rediscover SOD1 and TDP43, genes that we already knew about. So we knew the technique was working. But we also were able to identify TBK1 and NEK1 as two new ALS genes. And since then, other groups have gone on to use similar methodologies to find new genes for ALS. And we've gone on beyond that now, because we've got to get more sophisticated in how we do these analyses. And we now use something called regional collapsing, where instead of just asking which genes are affected, we're asking like microdomains, like this little tiny region of the gene. Show me the little tiny regions of the genes where ALS patients have mutations more than the cases. And I won't go into the details of this, but these are the different ways that we do it. And we're very excited to report that this also works. And so here's our most recent analysis, and LG-ALSL is the newest ALS gene. You heard it here first. It just came out last week in Genome Research, and it is actually telling us about an entirely unsuspected pathway, just as I was alluding to the importance of on the previous slide. So this actually has nothing to do with any of the other known ALS genes. It takes sugar moieties, called galectins, and it attaches them to proteins that have already been manufactured, and that pattern of galactonization tells the protein where to go. And so it's probably helping to act like the FedEx scanner to like ship proteins to where they need to go and get them to where they need to be delivered. And that's messed up, and we didn't know that that was happening in ALS until the discovery of this particular gene. So souping up the statistical analyses and the way that we look for these new genes is one approach. But I can tell you that the best way to discover new genes is to study more patients. And so really, more patients being sequenced and then numbers is exactly the way of the future and what needs to happen. And so I'm really happy that we've received funding from the ALS Association, as well as from Biogen, to create this study called Genomic Translation for ALS Care. And this is, when we first started the study 3 years ago, this title was somewhat aspirational. We thought, well, you know, someday we'll be able to translate the genome into care recommendations for patients. And I've already told you that for SOD1 and for C9orf72, that's on the horizon, if not already here. So it's no longer aspirational for some component of the population, and hopefully over time will become more and more the norm. So how does the GTAC study work? So basically, here are the participating centers. They're distributed across the United States, and then the entire country or region, however you want to think of it, of Scotland. So patients give us lots of blood. I'm sure that's not new to most patients in most ALS clinics, constantly being stuck and drawn. The DNA heads off to my laboratory. The RNA comes to me as well. We take future stem cells, so blood mononuclear cells that we can turn into stem cells in the future, and we put them in a repository at Cedars-Sinai. It's kind of a library of cells that we can go back to and create stem cells and study in the laboratory. And then a couple of basic lab tests that happen locally to kind of see where people are on some key features. Every 3 months at regular clinic visits or at research visits, we collect data from patients, including the exposures and epidemiological data that is almost identical to the one already being utilized by the CDC National Registry. So that as discoveries and findings come from the CDC, we can immediately turn back to our patients and look to see if there are environmental and gene interactions. And then unique to our study at the time when we started it was that we wanted patients who are participants to get the information back. So it used to be that when you did a research study like this, if you found something genetically, you weren't allowed to tell the individual. And I said, that's kind of ridiculous. I wasn't alone. There were other people who were starting to say things like that. And so we actually double-check every abnormality that we find that we think is causing the disease and use a genetic counselor to return that information back to participants and their families so that they can use that in their family planning and just to have as knowledge. So how's the study going? I need to update this slide as well because we just crossed our 1,000th patient mark. So this has been the fastest enrolling ALS study in the history of the world. And we're pretty proud of that. And that has taken a lot of hard work at places like OHSU. And I'm grateful to their staff who are here who have done a great job of recruiting patients from this region. And we've also made sure that we're working together with people who are doing similar studies in other places. And so in particular, Answer ALS and Project Mine have very similar platforms and similar ideas. And we're all working together because we realize that the more patients we've sequenced, the more genes we're going to discover, and we have to work together in order to get the most out of this. So I would say, and would wrap up today by saying that I think that there are many reasons for hope in 2019 for patients with ALS. So number one of the reasons, I guess I kind of have these out of order, I would say, is that researchers are working together as never before. And part of that comes from the ALS Ice Bucket Challenge money, right? As soon as scientists see blood in the water and some potential funding, they come swimming. And the ALS Association very wisely set up almost every dollar going out in research funding as collaborative work that basically brought people who used to be in their own silos working on their own together. And so now researchers are working together as never before. And the outpouring of that has been molecular therapies that are now nearing clinical use, particularly for SOD1, C9. And I know personally of 3 or 4 genetic gene replacement and knockdown studies that are about to get started. And then finally, new genes are emerging every few months, and I think that's telling us really valuable information about the pathways that are involved, as well as the individual targets that drug companies can go after. And so I was just at a conference for Target ALS, another group that funds research. And when that group started in 2009, there were 5 drugs in clinical development for ALS. And at this particular meeting, we heard about an additional 50 that are in the process. So there's been a 10-fold increase in the number of drugs that are being tested in animal models, cell models, and in humans. And hopefully we'll be able to use the emerging genetic information to take that ratio, 2 drugs out of 60 that we've tried, and increase it to where it should be, more on the order of 6 to 10. And who knows, maybe we'll get lucky and even do better than the average disease. So with that, I'll go ahead and pause and hand it over.
Thank you, Dr. Karam. We appreciate your time. And we'll have and just to remind everyone, because we're streaming this live, we're going to go through all of our presentations and have Q&A at the end with a full panel. So be sure to write down and jot down your questions and bring them back for all of our presenters. We're going to have more discussion about therapeutic developments for ALS, but there is also a very important aspect of the disease, which is about living with ALS. And we're thrilled today to have someone who is very experienced in understanding of exercise and in ALS. Dr. Eric Ensrud is the Associate Professor of Orthopedics and Rehabilitation at the School of Medicine at Oregon Health Sciences University, right here. He has a medical degree from University of Minnesota and did his residency at the Spaulding Rehabilitation Hospital at Harvard Medical School in Boston. He did fellowships in neuromuscular medicine and EMG fellowship at Brigham and Women's Hospital. He has a unique intersection between rehabilitation medicine and neurology, which is pretty rare. So we're thrilled to have Dr. Eric Ensrud join us. Welcome.
Thanks. Thanks for having me, Lance and Kathy and everyone, and thanks for asking me, Shafiq. I've known Shafiq for quite a while. And so this is something that comes up frequently with patients with questions about exercise. Is exercise bad? Is exercise good? And then I'll try and touch on an important aspect of musculoskeletal pain in the latter part of the talk. And I've actually put some handouts on your tables, so they've already been passed out about the most important part of musculoskeletal pain in ALS, which is the shoulder. I'm going to, Lance, if I'm going too long, wave your hands, because I've tried to cut these slides down, and then I'll try and get done in time here. So in exercise in ALS, we really have a basic question, and does exercise training, does it improve muscle strength, all these positive aspects, improve cardiorespiratory function, prevent disuse atrophy, and very importantly, improve overall quality of life or sense of well-being, or does exercise accelerate muscle breakdown, disease progression, and weakness in terms of an overuse syndrome? Or does it do both? And so one problem, whenever you talk about exercise and you're talking about exercise in someone with a medical condition, the word exercise, certainly in the United States, we tend to think of, this kind of meathead guy here lifting in the, this is a Planet Fitness commercial. If you haven't seen it, it's very funny because the guy says nothing but, I lift things up and put them down. And then you can see this Iron Woman athlete here who's obviously, exercising to a very intense level. But that really shortchanges exercise for most of us and really shortchanges the benefits. really exercise can be very helpful in a much lower intensity, like pool exercise or chair exercise. So we really need to broaden what we think of. Exercise isn't just about Jane Fonda or that P90X guy. So one thing to just make sure that we cover here today is the type of muscle contraction done with exercise. And these are terms that are used in describing the contraction of the muscle. This is the bicep here. And concentric contractions are where the muscle contracts and the muscle shortens. So he's pulling up on the dumbbell here, or she is, and the muscle gets shorter. But eccentric contractions are where the dumbbell's already up here, and the muscle is used to slow the rate of descent of that. So the muscle contracts while lengthening. And this is really important. I wanted to make sure I touched on it because eccentric contraction really causes injury to muscle. You can get delayed onset muscle soreness, and it elevates the CK, or creatine kinase, the muscle enzyme, which elevates with muscle damage. It causes a lot of strain on individual muscle fibers that can damage them. And the reason that it tends to be used a lot by people who exercise is that it can be very effective in normal muscle that has normal nerve supply. So you'll hear people say things like negatives or slow motion exercises. Let the weight slowly down. So there are quite a few benefits in exercise in patients who have neuromuscular disease, including ALS, and a significant one is the changes in what we call VO2 max, and that's the maximum rate of oxygen consumption, which equals fitness, and that can increase by quite a bit with exercise. And there are lots of physiologic reasons in your heart and circulatory system for that. There are also changes in the muscle itself with exercise. You get different levels of enzymes in the muscle. The mitochondria, which generate energy in the muscle, increase in size and number. And then really importantly, the blood supply to the muscle increases through capillaries, which increases blood flow to the muscle. And as a result of that, your skeletal muscle gets an increased ability to utilize oxygen. And there are lots of other important adaptations to exercise. And it increases endogenous opioid peptides, which helps with our sense of well-being. It can reduce pain. It's really important in terms of reducing depression, improving sleep quality, and in this case, particularly important with reducing contractures. And so, but when we talk about exercise and ALS specifically, there are definitely some clinical concerns. And one is overuse weakness. We've already got, the muscle is being affected by the decreased nerve supply from the disease which affects motor nerve cells. Is exercise a risk for ALS? I'm not going to solve that question today, but we'll talk a little bit about it, some of the evidence for that. And so, this came up with ALS really being more widely recognized with this famous athlete, Lou Gehrig, and Lou Gehrig's disease. There are lots of other famous athletes who have developed ALS. So some epidemiological evidence, or statistical evidence, suggests that vigorous physical activity in terms of heavy labor or competitive athletics may be a risk factor for ALS. There are quite a few papers on that. You can also find evidence when it's looked at systemically that there's no really conclusive evidence for physical activity as a risk factor for ALS. So what the evidence that it does occur, where there's definitely been some increased risk of developing ALS that's been reported among professional athletes, and we've all heard about the NFL, and many of us have heard about soccer. And people with ALS are more likely than people who don't have ALS to have participated in collegiate sports and have a thin body type. But we also have evidence to the contrary that physical activity is not associated, and again, a systemic review against that. So it's kind of mixed, but it kind of makes sense that very intense physical activity, you stress the motor nerve cells, which are these very long cells going out to the muscle, could be a risk factor. Here's a more recent one. This is interesting. This is at from Brigham and Women's Hospital. And just a huge study. They enrolled over 160,000 women. And they adjusted the odds ratio for ALS for people who did strenuous activity 3 or more days a week, compared with no reported strenuous physical activity. And there was about a 56% increased risk of ALS in the women in this study who did very strenuous physical activity 3 days or more per week. So this was the first study, certainly of this size, to report an increased risk for ALS mortality that was associated with strenuous physical activity in postmenopausal women. And, but they wanted to make sure they pointed out that this risk of ALS was much smaller than the overall benefit of strenuous physical activity for other serious conditions, including total mortality and heart disease and breast cancer. So the thing that we have here, these two cells, the white cell and the yellow cell, that are involved in ALS. The white cells are the upper motor neurons that go from the cortex down to the spinal cord, where they synapse or make a connection with these yellow cells, which are the anterior horn cells that go out to the muscle. These are very long cells, and they, because of that, have a lot of stress. Everything that's produced here has to be transported all the way down here. Everything that's produced in the nucleus or production center of the cell in the spinal cord has to be transported all the way out to the muscle. There's no connections in those cells. So we can find some evidence in animal models, and this was an important paper looking at exercise as an animal model of ALS, and these were mice who have the SOD1 gene, that first gene that was discovered in 1990 that Dr. Crum reported. This was 90 mice in each of 3 groups, mice that were sedentary, mice that were given moderate exercise, 30 minutes a day, 5 days a week, and mice that were given high exercise, 60 minutes per day, 5 days a week. And this device here, you can see the mice here. This is called a rotorod, and it's a way that you can assess physical activity in mice. The mice are put on the rod, and they don't want to fall down, so they'll keep running on the rod as it turns. And nothing bad happens to the mice when they fall down. I just wanted to make sure. I mean, there are lots of studies that bad things happen to mice, but not this one. Until the end of the study. So they measured the time that the mice are able to stay on this rotorod, and then the mice were sacrificed, and they looked at how many motor neurons do they have in the spinal cord. And this result was really interesting. So the mice in the moderate exercise group had significantly more motor neurons and more time on the rod than the sedentary or non-exercising mice or the high exercise group. The worst scores were in the mice that didn't do exercise. So this raises the idea, is there an evidence of a sweet spot in exercise where you don't exercise too much, but you are doing some exercise? And you can see here with the results here, this is the type of mouse, sedentary, moderate, or high intensity, and how dense are these motor neurons. So this is what they are in a wild-type mouse, 2.22, if they don't have the SOD1 gene mutation. If they're sedentary, it drops by more than half to 1.02. With moderate exercise, it's considerably better, 1.67. And then with high-intensity exercise, it's 1.43. So again, this evidence of a sweet spot for exercise. So let's look at some clinical trials, not in mice, but in real human beings who have ALS. This is a study from 2001 with 25 ALS patients, and they were randomized to an individual exercise program, 30 minutes a day, and they were assessed at 3, 6, 9, and 12 months. And the patients in the exercise group scored better on the ALS functional rating scale, and the Ashworth scale is a scale of spasticity, at 3 months compared to the controls, and a similar trend was seen at 6 months. Here's a second trial that's more recent from 2016. 60 ALS patients and 30 were randomized to one of 3 exercise groups. And in this trial, in each group did daily exercise for 2 weeks each month. The first group did an arm and leg ergometer. I'll have a picture of that, but that's a machine that you move like this or with the arms, and then an exercise bicycle. And then they did 3 repetition arm and exercise muscles if the muscle had a score on a rating scale that we use of greater than 3, which means that it can be contracted against the weight of the limb from gravity without a weight. And then a group that did just the exercise, and then a group that did passive range of motion. And this control group, in this case, did 2 days a week rather than daily passive exercise. So group number 1 that did the arm and leg ergometry, like the circular motion of the arms and legs, had significantly higher ALS FRS-R, which includes respiratory scores, at the end of the 6-month period. And interestingly, 6 months after the training done, they still had a higher score. And you can see that in this graph here. This is the ALS FRS score on the left, and this is the timeout. And this red here represents the difference between the group that did exercise and the group did not. And here's at 6 months. The ALS FRS scores have gone down, but there's still a considerable difference. And this difference was about 5, which is pretty good and is the equivalent or possibly a little better than Radicava, for example, which is effective as well. So it's not a minimal difference. This is the most recent published trial in 2018 from Clausen. This is the Mass General group on safety and tolerability of exercise in ALS. And importantly, this trial was studying whether exercise worsened ALS or whether there was no difference. It wasn't powered. They didn't have enough patients for efficacy. And they had 3 groups that did stretching or range of motion, resistance exercise, or endurance exercise, and they exercised for 6 months. And what they found is that all 3 groups were safe, as no difference in the rates of disease progression. Okay, so they're not over-exercising, and there's a trend towards fewer falls in resistance and exercise groups. This is the most recent paper. It's just a pilot study, but from just a couple of months ago, and that was recumbent stepping exercise in ALS, and that's using a machine like this. You can find them in most gyms, and that's ergometry, kind of like an elliptical of the feet with an elliptical of the hand. What was that guy, the Tony something that did that thing, the Gazelle or something? Do you remember that TV thing? A little bit like that. But this also found that this was safe and did not worsen patients. It's not powered to show an improvement. And I'll send this PowerPoint to Cassie so she could distribute it. We don't really have time to get into the details here, but this is the exercise protocol used in the Mass General study. And you can see they'll say like the number of reps done in 1 RM, that means 1 repetition maximum. So you see what the muscle can lift, and then you only do 40% or 50% or 70%. The important thing is that you're not over-exercising the muscle. And then this is their endurance exercise protocol. They did 10 minutes of upper limb exercise, followed by 10 minutes of lower limb exercise. And so we'll move on now to musculoskeletal pain in ALS, and I wanted to do a little practical teaching here, so that's why I have the handouts for you. What's most important? So a real interesting paper from the group at Leahy Clinic in Boston, Darlene Ho, in 2011, looked at shoulder pain and ALS, a survey, and 45%, nearly half of patients with ALS have experienced shoulder or neck pain. And in 2/3 of the shoulder pain patients, there was an unknown cause. In about 1 out of 5, they had a condition called adhesive capsulitis, where the shoulder capsule becomes inflamed and it's quite uncomfortable with motion. And then about 15% had rotator cuff tendinitis, where the main tendon of the rotator cuff. This is a really common condition. I'm sure there are multiple people in the audience here who have had rotator cuff tendinitis. What they found, though, that this is often undertreated. Only about 50% of the patients with ALS who had this had received treatment. And the really important things to try and prevent this and treat it are range of motion exercises, and then if those aren't successful, steroid injections can be helpful. This is just some shoulder anatomy, the way the bone, this is the upper arm coming into the scapula here, and this is with the muscles, and it's just a really complicated joint. It's the most complicated joint in the body, so when you get decreased muscle force in the muscles of the shoulder from the effect of ALS, you get an unbalance of that, and you can develop shoulder pain from that. So this publication from 2018 from the MGH group, this is with Sabrina Paganoni, who was a fellow with me with Shafiq. Boy, that was a great fellow class. They've both gone on to do great things working with patients with ALS. So this paper looked at exploring the use of educational material about shoulder dysfunction in ALS. And they gave this brochure to inform patients about what exercises to do for shoulder pain. And to start, this is the handout that you have on your table. And it has, so to start, these exercises are active when the patient can contract the muscle to make the movement. And the exercises, there are 3 exercises that are done 3 times each, and then hold on the edge of the range of motion for about 30 seconds and do those once or twice a day. And then once ALS has become significant enough that the muscle can't contract to go through that range of motion on its own, then these can be done with the assistance of a caregiver to do passive range of motion. And I would say the one thing about this paper is that they didn't put those pictures in the paper. So I'm going to have Shafiq come up here, and I'll show you. But it's three ranges. You know, the shoulder is such a real complicated joint because it can go through so many ranges of motion. So the first exercise that you do is shoulder flexion. Put your hand on and hold up like this until it won't go anymore. There's discomfort, and hold that for 30 seconds. You could have some mild discomfort. You don't want to get it to the point where it's super uncomfortable, okay? And back down. And do that 3 times, okay? And then the next exercise is to come out to the side and come out to the point where it's tight and causing some mild discomfort, and hold it there for 30 seconds. Okay. And back down, and then you repeat that 2 more times. And then the third time is to hold the elbow at the side and come out this way until it stops. Or you want to have the beginning of a little bit of discomfort, but not to where it hurts, and hold it for 30 seconds, and then come in like this. And you've got that on that handout that's been placed on your tables. It just shows the patient doing it themselves, and I think it's important to cover that these are just as effective, if not more effective, if someone's assisting with them. Thanks, Shafiq. Yep. So these are the exercises that you got there on the handout. So putting into practice, so getting started with exercise. Remember, one size doesn't fit all, and of course, some of these exercises as ALS advances are not possible, but we wanted to just cover that concentric exercises are good, eccentric exercises are not good. That possible exercise muscles with this muscle score MRC 4 or 5 or greater, that means that the muscle has some weakness but is greater than anti-gravity. And if you have questions about that, you can check with your neurologist because they're putting it in their exam notes, or your physical therapist, and why these muscles will respond to exercise. Once they get to just anti-gravity, they're probably getting enough exercise just from activities of daily living. And then focusing on these responsive muscles may enhance the overall function of limbs. I want to just make a point to avoid personal trainers. I have nothing against them. I've never had one myself, but I get one from my dad, who calls me a helicopter child. But the thing about personal trainers is that they're trained as a whole, as a group, they're trained on methods for normal muscle, and that the training doesn't include education about treating muscles with structural change or decreased nerve supply. and personal trainers tend to focus on eccentric exercises because they're very effective in normal muscle. And so they tend to push patients to full muscle fatigue. And so I have had patients with ALS who got personal trainers early in the stage of the disease, and it ended up being detrimental. Here's a curriculum for the Lake Washington Institute of Technology in Kirkland for a fitness specialist or personal trainer. And you can see here, they're learning a ton. And they're learning a ton about kinesiology, but none of this is about disease. Okay, so I would ask you to be So when you're doing aerobic exercise, you shouldn't do it to the point where you can't hold a comfortable conversation. That doesn't mean you have to talk the whole time. And I did make the mistake one time at Boston in telling a patient, you need to be able to talk the whole time. And she came back for a 6-month follow-up and said, it's going great. The exercise has gone well, but my family is sick of having to talk to me the whole time. I'm like, I need to explain myself better. So a real simple thing that you can do, the same thing they do in an exercise lab, is the talk test, where you're able to just speak comfortably during the exercise. You're exerting yourself, but you're not getting short enough of breath that you couldn't maintain a conversation. And there's good research that gets you to right where most of the exercise studies that are done in nerve and muscle disease are at. It's about 65% of VO2 max. And so we want you to think about doing stretching or range of motion exercises. They're really helpful for patients who have ALS to prevent contractures, reduce pain and spasticity. That gentle aerobic exercises, when possible, to maintain cardiovascular fitness can be performed as long as they're safe without a risk of falling or injury. And then you do it at a level that you can talk while exercising. And that moderate resistance strength training in earlier or mild ALS has shown to be helpful, and just don't overdo it. These are some, in early ALS, a weight that can be lifted 20 times. Do it about 8 to 10 times of that weight, so you're never going to full exhaustion of the muscle. And then later on in ALS, you can do a little more than half of what can be lifted one time, 3 sets of 3 reps, and again, to avoid personal trainers and communicate with physical therapists. It is the ALS Association of Southwestern Washington, so I put some Seahawk thing in there. So to make it real simple, no exercise is bad, moderate exercise is good, and extreme exercise is bad. And then, it really, in working with patients and giving them the go-ahead to exercise, I think it's really helpful for quality of life too, and the locus of control. An external locus of control, something happens to you, you can't really fight it. An internal locus of control, you can participate in the fight against the disease. And just a specific patient, a 65-year-old with ALS who I saw in Boston, a Vietnam vet, and he was self-referred in for a second opinion, and he'd been seen in Boston, a great ALS Association clinic. I may have quoted a paper from them today, but I won't say. But he was told by a physical therapist there, don't do any exercise, you're just going to make yourself weaker. And that was really discouraging to the patient and really added to the marked depression. It's just hard to sit here and take it. So, he had quite a bit of widespread decreased muscle force or weakness, but we counseled him about doing pool exercise and not overdoing it, because a lot of people who have ALS were really intense athletes. And so you have to tell them, don't push 100%. And at 3-month follow-up, he actually had gotten some increase in strength, and he just felt a lot better. I think his exact quote was, I'm going to give some fight to this bastard. At 6 months, the strength was no longer improved, but comparable to the initial assessment. At 9 months, the exam had worsened, but the affect was still better. Like how he felt was still better. So thanks. I hope I got done in time, and I just want to give special thanks to a couple of people who have done a lot of work in ALS and exercise. Thanks.
Okay, that's much better.
So we have some seated questions here from the audience that I'd like to start off with. And the first question is for Dr. Harms. You talked about looking at environmental exposure as part of this genetic aspect of the disease. One basic question is, what encompasses environmental exposures? What's being looked at? Not necessarily conclusive, but what are potential factors that are involved or being studied?
So the difficulty with studying environmental factors is that, while we all start off with the same 18 or 19,000 genes and have minor abnormalities that differ, we're 99.999% the same genetically. But if you think about how each of us lives our lives, they're probably 99% different. And so it's been hard to nail down exactly which environmental factors to look at. And so what we're looking at are the ones that have historically been looked at. But unless you think of what they are in advance and ask about them in advance, you can't get reliable or trustworthy data. And so that's a real difficulty in studying the environmental factors. So we're looking at pesticide exposure, solvent exposure, heavy metal exposure. We are collecting where everyone lived for their entire life so that we can do geolocation and try to estimate the exposure to toxic waste dumps or proximity to blue-green algae blooms in fresh water, some of the other things that have been looked at. But the difficulty will be, we have already a 3-hour assessment. So patients with ALS have to go through a grueling interview in order to get that data. And the more factors you look at, the longer that gets, the more expensive the study gets. And so it can be really challenging to pick which environmental factors are being assessed.
Great, thank you so much. This is for the clinicians, really. It's a question about clinical trial participation, and I'm going to combine two questions here. One person mentions that medical issues such as gallbladder surgeries have disqualified from clinical trials and studies. Another question talks about someone having ALS for more than 3 years. Are there opportunities for people to participate in clinical trials that who've had the disease longer or who have other types of medical conditions? What factors go into who gets to participate in trials?
So that's a great question, and we know that unfortunately most patients with ALS won't qualify for clinical trials. So it's almost always kind of the same group of patients who end up in a trial. And the major reason is that, as we mentioned, ALS is a heterogeneous disease, and people have many different reasons why they develop ALS. And the course of the disease of each single individual is very different from the other. And because of that, in order to kind of pick up a more homogeneous population, most of the clinical trialists, what they rely on is the inclusion and exclusion clinical criteria. I think that eventually, hopefully, we'll have different models to do clinical trials, but at this time, the best thing we have is to pick a homogeneous population to study them. So people who have longer than 3 years are a little bit of an outlier, and they may kind of skew the data and compared to people who are earlier in the disease and progress kind of at a faster pace. The question about the gallbladder, I think that was specifically regarding the Amylyx trial, and just because of the type of the medication, it's contraindicated in people who have already removed their gallbladder. So this depends really on the medication. That being said, I would like to also hear what Matt has to say about this.
So I would say that both of those things are very true, but there is some hope that we may be able to broaden who's involved in ALS studies in the future by using the prognostic models that Dr. Karam was showing. So if you could, at enrollment, predict where someone would be 6 months later accurately, then you could include anyone, no matter how long they'd had disease, and then you could just compare that patient to themselves on drug compared to what the computer said they would be like 6 months later. And so there are some computer algorithms and machine learning techniques that are trying to be used. And one of the outcomes of that could be broader inclusion of patients to be compared to their own control, themselves as a control.
Great. This is a question for Dr. Ensrud. Actually, I have two of them here. One, is it possible to hasten progression of ALS or to accelerate the death of motor neurons due to overexercising if one happens to overdo it?
The answer to that would be yes, based on the evidence that's available, and I would go back in particular to that mouse study that I showed, and the patients, the mice who did the extreme levels of exercising, which was a couple of hours a day, had greater death of motor neurons than patients who did no exercise or the moderate exercise.
And a follow-up question, very specific, but I think it's worth asking. This person does intense aerobic exercise twice a week, 30 minutes uphill biking, and in no way could hold a conversation. Is this not advisable? Does it help with lung function? Been diagnosed 9 months and have maintained this progress with only slight change in performance. No noticeable lasting effects.
Is that person, did they have the disease ALS?
Yes.
Or
Yes.
Yes. Well, if they're tolerating it well, you know, we've heard time and time again today about what a heterogeneous or widely spread presentation ALS has and how it's different in different patients and how there are a multitude of ever-expanding genes behind it. So I certainly wouldn't suggest for wide numbers of patients that intensity of exercise, but if a particular patient is tolerating that, I wouldn't tell them to stop, because they could potentially be receiving, you know, we talked about the benefits of exercise that go beyond muscle strength and so on, endogenous opioids, a sense of well-being. I suspect anyone who exercises at that intensity has what I call a high motor drive and really needs to do that and won't feel well if they don't.
Great, thank you so much. This is for the whole panel. The mouse model and stem cells seem to be being used for testing therapies. Have there been new mouse models developed, or is all new testing going to be on stem cells?
So I'll take a stab at it. There's a lot of interest in moving to stem cells, but there's also a lot of limitations with it. People say we've differentiated the cell into a motor neuron, but it's basically they have a few characteristics. So it's good for screening. The power of genetics is actually so much better than it was that you can learn a lot that way and compare to what's going on in humans. But there's also an enormous amount of work going on making other mouse models and also other transgenic animals. So while the success of creating a model that's quite as robust as the SOD mouse hasn't happened, there's still a lot of work or a lot to be discovered in it. So someone mentioned that you shouldn't talk about trial failures, but it's trials that didn't work. The same is true of the mouse models. You learn an enormous amount about it, even though you didn't get motor neurons necessarily to die. And in some ways, you can learn a lot about what else is happening in the cells that surround it. So there's a huge need to keep going with animal models, and there's still a lot of potential for doing stem cells. And there's really a lot of groups doing this in different ways.
And I think I would add that no one is just screening in a single model anymore. So people will look in the SOD1 mouse and one of the TDP43 mice and the FUS mouse and a zebrafish, or they'll look at the human stem cells and the SOD1. So everyone is kind of cross-calibrating different drug screening to make sure that we're not seeing an effect of just utilizing kind of one different test. So you'll get better, potentially you'll get better, more reliable information by bringing together things that worked in multiple different models.
Great, thank you so much. This is a question for Dr. Karam. Are there any studies looking at cumulative effects of taking multiple supplements together?
So one of I'm not aware of any study, but I know that, for example, one of the protocol called Diana protocol, which was made by a doctor who's, I believe, their daughter or son had ALS, and was a combination of multiple different vitamins and supplements. And if you look at each compound, some of them, again, looking at the ALS Untangled, may have some effect, and some other may have actually some negative effect potentially. So, but I'm personally not aware of any clinical trial looking at multiple supplements and seeing whether they would work or not. Matt, do you? No.
Okay. Next question. Is it possible for significant genes implicated in ALS to cause other genes to mutate negatively and impact ALS progression?
Yeah, so that's a really good question. So the vast majority of ALS gene mutations we don't know of having a role in generating additional mutations. There are some studies underway to look at DNA damage in the cells of patients who have ALS. And so that's actually something to pay attention to in the coming years, because you could envision that one of the other potential ways that sporadic ALS could occur is that you don't actually have a DNA mutation at birth, but then as cells divide and multiply and grow over the course of your life, a mutation could pop up in what I would call kind of a seed cell. And so that one cell, maybe a half a dozen cells, could have a mutation that arose spontaneously during development, and then it could kind of trigger the process that would spread from cell to cell and create ALS. And so that's something to pay attention to, but we don't know of any yet. Some of the genes that are mutated in ALS have a function in maintaining DNA, but we haven't been able yet to establish a link that the mutations themselves go on to create more DNA damage.
Another genetics question, which is genes regulate proteins. Why are these genes affecting motor neurons and not sensory neurons?
So that gets to kind of what in the ALS research community we call selective vulnerability. So it's a very good question. Why is it that motor neurons are more susceptible to ALS gene mutations? That question is being asked kind of across all neurodegenerative disease. So all of the proteins that are causing Alzheimer's disease are expressed everywhere, and yet they affect the memory neurons. The ones that cause Parkinson's disease are expressed everywhere, but they affect the ones in the basal ganglia and in portions of the brainstem that give rise to Parkinson's disease. So this is a general question that we don't have a great answer to at this moment. But it is an area of intense research that I think will be very productive, because if I could figure out why a sensory neuron is protected, then I could try to mimic that in the motor neurons to protect the motor neurons. Or if I could figure out why it is that motor neurons are susceptible, we could kind of compensate for that and kind of increase the number of compensations to try to prevent the neuron from getting sick or delaying how long it's healthy.
So I'd like to expand on that. There is actually a major difference between sensory neurons and motor neurons. And motor neurons are much larger, and they're metabolically much more active, and they have a lot of mitochondria to produce more energy. And they also, there are fewer of them, and they control only a few muscles. But this is where this idea of protecting mitochondria and some of the related processes are really important. So that, and why these therapies might be having a much broader effect than what people anticipated, because if you can keep the mitochondria working better, you can protect these cells. So a key idea in mitochondria is something called reserve capacity, that mitochondria have a lot of excess capability to produce energy and to do the other things it needs to do, and it wears down in the disease. And many of these mutations that have been identified do affect the way mitochondria function to get proteins in. And the key point is when you cross that threshold where they can't meet the needs anymore. And so that's where if you can protect the mitochondria a little bit, you can push it above that threshold, and it keeps the motor neuron alive much longer. And there are reasons to suspect why motor neurons are really susceptible in that way. It's also why a lot of environmental toxins might affect motor neurons more than other neurons.
That said, there are kind of sub-questions because there are some motor neurons that do quite well in the face of ALS and are preserved. The ones that run eyeballs, for example, the ones that control the external anal sphincter, and then even within the spinal cord at levels where the motor neurons are affected, there are some subclasses of motor neurons that are much less susceptible to the disease also. So there are a lot of really interesting things to be figured out that I think will have major implications for drugs.
And if you look at those subclasses, the ones that first die quite often are fast twitch, the motor neurons that work fastest and most reactive. And then the ones that are slow, that basically use for posture, those tend to be more protected. So you can see classes of where motor neurons start to die in these different models. But yeah, there's just a ton of possibilities and things we are just beginning to really understand the differences, and a lot that you learn by studying it.
Great. So more questions about genetics. I think that's a hot topic, as always. Can the panel comment on the availability of genetic testing for just people living with ALS today, and would you recommend that for patients, or what's the value or what's the risk in it?
So 6 months ago, the major reason to get genetic testing was for personal knowledge. You would find out you had a gene mutation, and that would influence kind of maybe how your kids thought about their own personal health or the advisability of reproduction. It even might inform how you reproduce. So it's now possible to do pre-implantation genetic testing so that embryos that have been fertilized that don't carry the mutation can be implanted, and so you skip passing it on. So that was up till 6 months ago. Now that there are molecularly directed clinical therapies, many of my patients want to be tested to know whether they're eligible for those clinical trials for SOD1 or for the C9orf72 gene. And actually, clinical testing has gotten a lot more cost effective. So in our clinic, if you want to get C9orf72 tested, there are a number of labs that offer that for $250, even if you end up having to pay for it yourself. Which happens probably 10% of the time. For most of our patients, we can get the insurance company to cover C9 because it's so prevalent. And then there's a company at the moment called Invitae Genetics, and I have no financial connection to them, but they are trying to undercut the market for sequencing. And so they have an ALS gene panel with 24 genes, and that's also a $250 out-of-pocket cost. So it used to cost $17,000 to do those 24 genes. Now it's down to $250. And so I'd say that 80% of my clinic population ends up having both of those things sent. And obviously, $500 is nothing to sneeze at. That's can be a significant financial burden. But people at Columbia joke that it's actually less than parking in our parking garage. But anyway, the genetic testing is much more ubiquitous, and and if that's not possible, then enrolling in a number of different clinical trials like the GTAC study, where it's provided for free, is another possibility.
Follow-up question to that: for genetic testing and studies, do you ever advise genetic testing or involve in the study siblings, surviving spouses, family members, and what can those people expect to? Is it advisable for family members to do genetic testing?
Right. So one of the things that we know for sure is that if these antisense oligonucleotides work for C9orf72 and SOD1 or other genes, they are likely to work best if you get them into a person before symptoms begin. So by the time someone develops weakness from ALS, in a hand, for example, we know that half of the motor neurons in the spinal cord at that area have already died off. So we want to be able to anticipate who is going to get symptoms and of the gene carriers when they're going to get symptoms. So there are now 3 studies in the United States and more in Europe where we are involving the first-degree relatives of people who are known carriers of the gene mutation testing them to find out whether they're carrying the gene or not. And if they're carrying the gene, following them every 6 months, doing clinical exams, believe it or not, doing an EMG every 6 months, a spinal tap every 6 months to look in their spinal fluid, to be able to look at protein markers to try to predict when somebody who's carrying the gene will convert from being asymptomatic, no symptoms of ALS, to developing symptoms of ALS, so that we could basically say, like you can in Alzheimer's disease, oh, hey, a year before people develop symptoms, this protein marker starts to go up in the spinal fluid, and that's when you'd start the drug. So, so people feel very differently about whether you should just get tested to know whether you carry the gene, but I would argue that you should at least consider it to know whether you can help participate in research that will help us in the future be able to predict who's going to develop symptoms, when they're going to develop them, so that we can get those drugs into people earlier. So it's a very nuanced question that you should discuss with your doctor or a genetic counselor. There are some potential theoretical risks to insurability and things like that if you do get tested and find out that you have the gene. There are potential psychological ramifications if you find out that you have it, or sometimes even if you find out that you don't, but your sibling does, there can be a survivor guilt. So it's a very nuanced question, but one worth having a discussion with a genetic counselor or your physician about.
Great, thank you so much. Follow-up questions probably for Dr. Karam and Dr. Harms about the GTAC study. First, are there any limitations for enrolling? Can a person have a short prognosis and enroll in the study? And then the second follow-up question is how do you enroll in the study? And if you shared that you share the results, how are those results shared with people who participate?
Sure. So we intentionally set the inclusion criteria, made minimal inclusion criteria. People who are unlikely to be able to return for more than one visit are excluded, because we need a second time point to predict the rate of decline in the vital capacity and the ALS-FRS. For that reason, people who are already on full-time ventilation can't participate. But other than that, you can have primary lateral sclerosis, progressive muscular atrophy, ALS, and as long as you're over the age of 18, you can participate. The one thing I would say is that it does take us between 6 and 9 months to do the whole genome sequencing and to get the results. So if you have a short prognosis, don't rely on participating in a study like GTAC to get usable genetic information in time. So clinical genetic testing would be much more effective at that, because the turnaround time is more on the order of a month as opposed to the 6 to 9 months it takes within the study. And then in terms of how to contact OHSU for possible participation.
So to participate, you don't have to be a patient in our clinic. So you can be at Providence or Kaiser or anywhere else. All you have to do is just contact our ALS nurse, Ivel, and she's there, so she can give you her contact information. But also, if you just Google OHSU ALS, you can have the contact information for Ivel or Diana Dimitrova, who is also a research coordinator, and she has her email actually on the website I just found out today. So if you're interested, please do it. But as Dr. Harms just said, if you want to know quickly, then the commercial available genetic testing are the way to go.
Great, thank you. This is a question for Dr. Beckman. Copper ATSM looks like a potentially promising treatment being studied. Will there be an opportunity for a United States trial of copper ATSM?
So almost certainly there will be, but it's still a large challenge of where do you raise the money. And the Australians were actually really interested in moving forward. So the idea is you could do the study in Australia, then move it to the States. It's one of the things that just where you talk about the complications, there's confidentiality rules I can't talk about, but there are a lot of different pieces and things being tried and different questions that come up that have to be answered before the trial would be moved forward. So the story seems to shift every couple of days but definitely it's attracting a lot of interest in the forced vital capacity results I think are really creating a lot more interest in moving it quicker here.
A follow-up question. Copper ATSM, along with several other treatments, have been a focus of right-to-try efforts for the idea that people with a terminal diagnosis like ALS should be able to have early access to potential therapies. For the panel, what is your perspective as clinicians in terms of right to try, and are there opportunities for people to get access to experimental medications before FDA approval?
So maybe I'll start. So one of my partners at Columbia sits on an ethics committee that's formed from lots of different universities that talk about this on a regular basis. And maybe it's just useful to briefly spell out what the right to try law is and how that differs from compassionate use mechanisms that were already in existence before the law. So it's always been the case that a university-based physician typically can petition the FDA for early access or compassionate use access to a drug that has already been tested in humans. As long as there's safety data, you could ask the institutional review ethics committee at your university to approve a limited study for a single patient or a small group of patients to get access to the drug. In order to do that, you had to have the buy-in of the pharmaceutical company to provide the drug in order to give it to you. So the right to try law changes very little about that. It builds in some protections, some legal protections for universities and drug companies that would do this. For example, if a pharmaceutical company gives the drug to an individual for right to try and it kills them, the FDA could not then use that against the company for a future use of that drug and say, well, but look, it killed that patient, the safety data is terrible, and that sort of thing. And it protects the physicians and the nurses who administer this drug that may not be safe in humans, legal protections so that you can't be sued if something goes wrong during the administration. And so I think the right to try law, unfortunately, doesn't actually do that much. It sounds very innovative and it sounds very pro-patient and advocacy-oriented, but it hasn't really moved the bar that much, and we're still in the same space where a pharmaceutical company has to be willing to provide the drug, and you have to have some safety data to make sure that it's ethical to administer it. So I'm very much in favor of the FDA's existing compassionate use program. I don't think the right to try law changes that much about it, but do recognize that there are lots of situations where early access gets in the way of what I would call definitive studies, right? So if it's very expensive to manufacture the drug and there's $1 million worth of it left, if the drug company agrees to give that to 10 patients outside of a trial and uses up that $1 million, then that $1 million is not available for a carefully controlled randomized trial that would actually tell us whether it worked or not. And so it does kind of get into the finances of it. If there were unlimited drug supplies and unlimited manpower to follow patients and that sort of thing, then it would make a lot of sense. But that's not the reality. I'll also add that there are some types of ALS that are so rare that it would be impossible to do a clinical trial. So some of you may have seen the press around the J.C. Hermstad case, where she has a unique mutation, and there happens to be a drug company that had developed a very early stage molecule that might tackle that one type of gene problem, and the FDA did allow for an early access compassionate use administration in that situation because there would never potentially even be a clinical trial. It's just too rare. So I think the FDA is becoming more open to these types of things, and we'll see where it goes.
Speaking of innovation in testing treatments, there's lots of discussion really about the value and the need for placebo groups in ALS clinical trials. Could the panel discuss, are there alternative trial models that could give greater access to potential treatments?
That's a great question. I think that it depends on what you're studying. If you have a drug that cures, that your hope is that it will stop the progression of ALS, and I think you can do a trial where everybody gets the drug, and if the progression of the disease stops, then there you go, you have your answer. Unfortunately, I don't think we have a compound like this at this time, for sure. And then, so most of the drugs that we are studying are medication that will kind of slow down the progression of the disease. And when you do a study, you're already pre-selecting certain patients, so you're already biased. You're not taking everybody. And then there's a placebo effect. So you're already selecting patients that probably have a little bit higher response to placebo effect. And if you're not having a control group, and you're just giving them the medication, they may feel a little bit better. They may slow down a little bit the progression of the disease. And not only that, we know that with ALS, the trajectory is not always linear. So some patient kind of for several months kind of plateau, or their ALS-FRS score, which is one most of the trial look at, improves a little bit. So it would really be impossible to tell whether a drug is really working or not. So I think that's the reason why we do these kind of randomized trial with a placebo, and everybody is blinded to whether the patient is taking the placebo. So the patient doesn't know whether they're taking drug or placebo, and the doctor and the pharmacist and the nurse administering the drug don't know what the patient are getting either. The other way to kind of be nicer about this is to change the ratio of placebo versus real drug. So for example, some of the trial that we do, you have 2 in 3 chances of getting the drug versus 1 in 3 chances of getting placebo, or even 3 chances out of 4 in getting the drug and 1 chance out of 4 in getting placebo. So it really depends on who's designing the trial and what they're looking at. Another way to look at things is with biomarkers. So, with SOD1, you are able to measure the SOD1 protein in the CSF. So that's great. So that kind of gives us an indication that a drug may actually work or not. And then I had a third thing I wanted to say, but I completely forgot.
So I'd like to add to this, if it's okay, that the way the FDA regulates drugs differs for different groups. So the way you approach cancer is a group of experts that deals with that disease. And with neurology, there's a different set of panels, and the diseases are different, and there's expertise. But there's also some ways of taking trial design between the two that are being explored. And one of the things that's being done is to start to look at neurological diseases where you use the same group of placebos with multiple groups of different treatments. So there's really no way to know that there's a drug that's going to cure ALS that's being kept away. The expectation is a lot of them are going to fail like the other 60 before or have a very slight effect. But the idea is to use this with multiple centers and randomization where you could be on a different drug than other groups, but at least you're on something that has an expectation of working, and the chances of being on a placebo are lower. And so there's a lot of work going on in getting that up and running in the near future.
Like really near future, like this year, a platform trial run by the Northeast ALS and MGH Healy Center should come online, and it's doing just that. So there'll be 3 treatment arms and a shared placebo group. So you'll have an increased chance of being on a drug because you only have to have a small group shared amongst all of them on the placebo.
And Dr. Crum and Dr. Harms, can you share about the NEALS group, what it is, and how that disseminates across the whole country?
Yeah, so the Northeast ALS Consortium is actually no longer just the Northeast. It actually includes all of North America, Canada, and then there are sites internationally as well. There's been, hubbub forever about whether it should change its name to more accurately reflect that. But regardless, it was a group that basically came together and said, we need to have a bunch of clinical centers that are already geared up, trained, experienced in clinical trials in order to do ALS studies more efficiently. And so really it's a clearinghouse where drug companies come to get input on the design of their drug. They meet with the scientific advisory board to pitch new drugs and new targets to see what experts think about it. Then there's a set of experts that are skilled in the ins and outs of designing the clinical trials. And then when it comes time to actually launch one, the NEALS consortium of centers are already staffed with physicians, nurses, physical therapy therapists who do different types of evaluations and know how to collect the data that's necessary for a clinical trial so that it can more effectively and more efficiently, like for less money, do the clinical studies.
Yeah, and I would just share that both Oregon Health Sciences University and the Providence ALS Center are both very active in the NEALS consortium. This also is a question that goes a little bit towards Dr. Beckman and the clinicians on the panel. Dr. Beckman, when the SOD in my study had a 25% improvement of life expectancy, did any of the mice regain some muscle control or any improvement in their physical capabilities?
So the mice that had a 1 month extension of life, or 5 weeks, which was considered pretty remarkable, their decline was about the same as the other mice. They just survived longer. We worked on modifying the mouse model, and I didn't try to explain it, but SOD is a protein that has a chaperone, or another protein that helps it fold and pick up copper. And when you put the human chaperone gene in with the copper SOD, the mice actually die 8 times faster. In those mice, we could actually give the drug and keep them alive. We could take the drug away, and 2 to 3 months later, they start to develop the disease, and we could give the drug again. And when you gave the drug to those mice, they actually started to improve in function. They gained body weight, and they did physically look much better. They eventually died of disease, and faster than if they were on the drug the whole time. And so it really gave a way of turning the model on and off. And unfortunately, no one has actually bothered to follow up on those studies. You know, it's considered a strange model that nobody wants to do. And it's expensive to do because you have to breed 2 sets of mice and cross them, and then you have to work with them very early on. So you have to be in the vivarium working with the mice 7 days a week and twice a day. So there's tedious studies.
So following along that question, are there opportunities to look at treatments that improve function? This question is for people living with ALS. Are there any trials being done to restore function, or is it just to stop progression?
So I guess that's something that we haven't talked a lot about, and that's kind of technological advancements to improve the lives of patients with ALS. You know, we're mostly relying at this point on slowing down the disease to maintain function, but there's a whole set of biomedical engineers that are developing amazing things to help restore function. You know, exoskeletons to restore gait and walking, robots that assist with feeding or restore some limited hand function, better and better eye gaze technology that allows you to communicate faster through your iPad. At Columbia, we have a group that's using piezoelectric and 3D printing to create a dynamic head brace that basically would support the head, and just by a tiny little twitch in the direction you want to go, tell the computer to help you turn your head all that direction. And so technology that's allowing the miniaturization of motors and electronic computer chips to do the processing are also, I think, poised to make a real difference in terms of augmenting the function that people are losing. And so that's another very exciting area of ALS research that is, I think, on the cusp of making some real strides. And
yeah,
so,
I think Follow-up question, Dr. Ensrud, are there a lot of clinical management research studies being done that look at similar issues?
There is a group at Brown University that's been looking at implantation of electrodes to then power interaction in muscle. But it would largely be taking what motor function is available and enhancing it, like Matt was mentioning.
I guess so. In terms of clinical practice management, I think one of the exciting things is that as the electronic medical record develops, there's an opportunity for comparisons between the different types of care that are being provided at different clinics to find what types of care and when they're provided and how they're provided influence outcomes. So this is an area of research called patient-centered outcome-related research, or PCORI for short. And the way it works basically is, everyone who's on EPIC, the electronic medical record, the information about patients at each of their clinical trials gets anonymized, but then put into a central database. And then you can actually compare across 100 different ALS clinics to find out which ones are having the best outcomes, and then look back at what they're doing at each of the clinic visits, how they're doing it, to basically figure out what best practices are that really have noticeable influences on outcomes. And then there are individual studies, like the Eat More study that's looking to find out what types of diets may be playing a role in slowing down the progression. There are different types of exercise studies still ongoing to try to provide guidance on that. Different studies of swallow function to figure out what types of exercises might prolong the ability to swallow. So yeah, there are a lot of studies going on in that space as well.
This is about a specific clinical trial. Is there any opportunity for expansion of the Brainstorm trial from the Israeli company, and is there potential opportunity for that to be in the Pacific Northwest?
So the short answer is no, and they'll probably get to the patient that they need to enroll within, I think, August this year. So they don't have a lot of centers, which is sad, and that's why some of our patients have to travel to California, and they don't know if they're getting the drug or they're getting placebo. So hopefully we'll get the results, in mid-2020, and maybe, if it's positive, then it will be available to patients.
So one of the difficulties is, for Brainstorm, you have a bone marrow biopsy, and then those cells have to be grown in a Petri dish, and proximity to the labs where that Petri dish growing takes place is mandatory at this point for the technology. And so one of the locations of the sites are pretty much determined where the laboratories can produce the cells. And so unless the infrastructure for growing the cells expands, the number of sites that are close enough can't expand. But if it works, guaranteed they'll find a way.
Great. We're close to the end of our time. I know our panel is going to be available to talk with folks in the audience who have additional questions about their circumstances or about their scientific interests. But in closing, for Dr. Harms and the whole panel, really, talking about the pace at which and the resources it takes to advance studies. Dr. Harm, you mentioned that there may be 100 genes out there that need to be identified. Is there any way to predict the amount of time it will take and resources it will take to identify those genes? And for the whole panel, what's it going to take to move the community towards more treatments?
So I'm a glass half full kind of guy. And so I think that I may be underestimating, is I guess what I'm saying. So the entire GTAC project to study 1,500 individuals is costing $7 million. So we've estimated by doing calculations that we actually need on the order of 30,000 individuals. So we're talking another ice bucket challenge to identify those 100 genes, and then the pace at which we can do whole genomes. My laboratory back at the Institute for Genomic Medicine can sequence 100 genomes in a weekend. So it's really not a limitation of the machines anymore. It's a limitation of collecting the patient's DNA and then having the money to pay for it. So I would say $100 million and 5 years, we could find the additional genes. That says nothing of the much more arduous task of figuring out how the genes are acting, how the mutations are affecting them, designing the treatments that might help those genes, and then moving those forward into trials, which is an order of magnitude harder and more expensive.
Just to be a little bit more optimistic, you mentioned that 2 years ago it would cost $17,000 to do a panel, and now it costs you $25, $20.
Right. So the cost of the machines have become negligible. Now the cost is the salary support for the postdoctoral candidates and the people who are actually running the computers. The computing part is a negligible component of that, but a graduate student gets paid $35,000, a postdoc between $50 and $75, and in New York it's a lot higher than that. And so really the costs are now human costs, and there's almost no way around that component.
Well, thank you all so much. I appreciate all your time and your energy. I'd like to just say it's exciting to me to hear the work of our panelists, both with working directly with people living with ALS, but also in the bench science that leads to new potential therapies. These are 4 people from a snapshot of hundreds and hundreds or even thousands who are turning their eyes towards figuring out the puzzle of what ALS is. And I'm encouraged because of the pace at which change. I think Dr. Crom talked about technology and energy and effort is how we're going to move forward. And I'm excited to have brilliant minds like these working on the problems. So we want to thank all of you for being part of our panel today, and let's give them a round of applause. They're going to be here for a few minutes if you want to approach them and ask them personal questions. I want to encourage everyone to visit all the different tables and the ALS clinical trials tables that are here. I'd also like to just encourage everyone to engage with us with the ALS Association. Our hope is to create community that both supports research and helps promote the well-being and living well of people with ALS. And encourage everyone particularly to join us this fall at a Walk to Defeat ALS near you. So thank you, everybody.